ठीक है. Shweta, can you let people in the waiting room?
Yes, correct.
Great. We are live. I'll keep checking YouTube. Dispatches पे भी हो जाएगा. I'll check it. Just let everyone. Shweta, there's an admit all button. No? Shweta, there's an admit all on top. Just admit all in one go. That's easier. Who do we see? I see Satyam. Hi, Satyam. Good morning. You're the first speaker today morning.
Hi,
Welcome.
Chris. Thank you. Thank you.
Hope you're
Thank you.
feeling. Shweta, just mute someone, Shweta. Somebody needs to be muted. Hi, hi, Kiran. Hi, Ram. So good to see you both. We'll start at 10 sharp. I have a few slides of introduction. And then Satyam, you'll be ready with your slides to share. Satyam, you are ready with your slides now?
Yes, ma'am. Sure.
Great. Great. Hi, Ram. Hi, Kiran. Please meet my colleagues at India Bioscience. Shweta,
Hello.
Ankita, Arushi, and Rituja. Hi, Kiran. Hi, Ram. Thanks for making time for this.
Sure, no problem. Yeah.
Great. It's been going well so far, and quite a lot of turnout in the audience. The speakers have been doing a great job. And we have people coming in and coming out through the hour, through the session.
Okay.
Yeah. So,
I guess
yeah,
we'll
that's.
wait for a few more minutes.
Yeah, couple of minutes. We'll start in a couple of minutes, Kiran. You can put your video off and all till then. We'll let you know. Yeah.
Yeah.
Yeah. Hi, Debraj.
Hi Karishma, how are you?
Doing great. You're on the other side, Debraj.
Yeah,
What's that like?
I think we'll see through it.
Great, Debraj. Great.
Yeah.
Great. Welcome. All the best. You're speaker number 3 now.
Thank you. Yeah.
That's right. Yeah. Speaker number 2 is Sweetie. I don't see her yet. She probably joined. Ankita, we put, yes, please, Ram.
No, you can hear me. I just wanted to confirm that.
Yes. Yes, we can. Yeah. Ankita, we put a social media post now in the morning. Okay. So, Satyam, I'm assuming, Shweta, waiting room. Huh? Satyam, I'm assuming 14 slides and no more.
Yeah. I have 14 slides only. Yeah.
Great. Yesterday we had a bit of a record. We got 32, one of our speakers had 32 slides in 20 minutes. I was like, whoa,
Oh my goodness.
that's interesting. They were great talks. All of them were great talks.
Yeah, I saw on YouTube, so they were
Yeah.
amazing talks actually.
Really good talks.
Yep.
Hey, Karishma, can you just very briefly, maybe all members are here, all committee? They're
Not
all-
yet. Aarti is also the third member, Aarti Ramesh. I don't see her yet.
Okay.
Yeah, I'm wrong, but I'll just quickly check. No, as far as I see, I don't see her.
I guess you will tell us what we need to do, right? I mean, I kind of know, but
Yes, Kiran. Basically, Kiran and Ram, what you need to do is listen to the talks, which will be fun because they're really good talks we've shortlisted. And then 20 minutes there. Each speaker has been given 20 minutes. And in 10 minutes of expert mentor, question, answer, suggestions, feedback. They don't just have to be questions. They could all be suggestions on content, presentation style, how to make graphs, whatever you observed from the talk. And that's in 10 minutes. And if after that, after your inputs, we have time, then we will open it to audience Q&A. But if there's no time, we'll move to the next speaker, and audience can keep typing their questions in the chat window. So that's it.
Okay.
So, Kiran, we have 5 speakers in molecular biology.
Okay.
Yeah. And molecular biology, cellular biology were high demand. We struggled to make the shortlist because of lots of applications.
Yeah.
These promise to be really good talks. Yeah. Great. We're still having people come. Okay, it's 10. Manjula, I just had a request for the session. Thanks for being here. Good morning. Um,
Good morning, Karishma.
good morning, Manjula. Manjula, couple of things. We sort of have a few changes in roles. Ek to, screenshots as always. You just take screenshots. And Manjula, can you manage time, please? When I say start 20 minutes, just let us know when 20 minutes are up. Does that sound fine? Yeah, great. Okay, let me know if any questions in the chat window. So 20 minutes for talk and 10 minutes for Q&A. Okay, shall I start? Arushi? Hi, Aarti. Good morning. Nice to see you.
Morning. Good morning.
Hi, nice to see you. Yeah, welcome, Aarti. And Aarti, our other expert mentors are Kiran and Ram. You may know them.
Can you hear me?
Little connectivity issue, Aarti. Little connectivity issue. But now we can hear you. Yeah.
I can't. I don't know.
Yeah, okay. Yes, Aarti. Hi. Okay, we'll get
Hi,
started,
hi.
Aarti. Yeah.
Yeah,
Hi.
please.
Sure. So just a quick check. Please do keep an eye on the waiting room, and once the speaker starts, we'll count 20 minutes. Okay, welcome everybody. I hope you can hear, see us. Welcome to PhD Cafe. We are on day 3, session 5. Yeah, I've sort of managed to keep track so far. Okay, so welcome. Thank you all for your time. Speakers and expert mentors, as well as Team India Bioscience. And speakers, thank you for sharing your science with us in advance. We've been really enjoying the sessions. This has been a science fest. So this is what's lined up in molecular biology for the day. Lots of applications. We shortlisted you all, and 5 of you are going to be telling us about your work. We are really excited about this. So all speakers will have 20 minutes to give their talk. Please do stick to time because we are tight. And then we have 10 minutes for Q&A. And the Q&A will be led by our expert mentors, Kiran, Ram, and Aarti. They can give you suggestions, feedback, and so on. And if we have time, we will take audience Q&A, which we've been doing through the last 2 days. But the audience can always use the chat window to type their questions. And it's not just because we want to hear from you, but because we are incentivizing hearing from you by saying we have a giveaway alert, which means that the audience member who has been keeping the chat window most active with their questions, suggestions, feedback have been awarded a physical copy of this new e-compendium from India Bioscience, which was led by my colleague Ankita. And it's about challenges and opportunities in life science PhDs in India. So it's hot off the press. E-compendium was just printed and published, and you get a chance to get a free copy. So we've had 4 winners so far, and today's session, we are in line for the 5th winner. Great. So thank you so much for our expert mentors for your time. General instructions, hear us, see the slides. Use the chat window, keep it active. 20 minutes plus 10 minutes. And of course, feedback should be constructive to better the science, in the correct spirit of giving feedback. All right, after this at 1 p.m., when the session ends, we have an open networking session with Snehal Kadam, who's finished her PhD recently from Hull York Medical School. She's going to be fielding questions, open session on building professional networks as a PhD researcher. Different meeting link. This link directly leads you to that meeting room. PhD researchers see you there at 1 p.m. But between now and 1 p.m., lots of fun science is going to happen, and it's going to be led by Satyam Singh. Satyam, thanks so much for joining the meeting room early, and let's get started.
Thank you, Karishma. Thank you, Karishma, for your nice introduction. And I would like to thank India Bioscience for this great opportunity. So I'm going to begin my presentation. So my PhD work, I actually work
Satyam,
on.
start sharing slides. Sorry to interrupt. Start sharing slides.
So I have already sharing the slides.
No,
Can you
we can't see. No,
I
maybe I
can
can.
see. Yeah.
Okay, I'm so sorry, Satyam. So sorry. Go ahead. Go ahead.
Okay, thank you. So, my PhD work actually, where I have worked on the development of a novel alkyl AG3 derivatives, which is a green tea polyphenol-based compound, as a third generation EGFR tyrosine kinase inhibitors to overcome from the drug resistance for the cancer treatment. So before starting, I would like to give a brief introduction about EGFR, which is also known as epidermal growth factor receptor, which is mainly found on the surface of the outer cell membrane. And upon binding of the ligands such as EGF, TGF alpha, or amphiroglin, these EGFR protein undergoes homo or heterodimerization. And upon homo and heterodimerization, these EGFR protein activate the downstream signaling pathway, which is phospho-AKT and RAS-RAF-MAPK pathway. And these pathways are very crucial for cell proliferation, cell survival. And it has been found that EGFR overexpression is actively associated with the poor prognosis of the lung cancer, breast cancer, melanoma, glioblastoma, etc. At present, there are two types of therapeutic approaches are available to combat this EGFR-mediated cancer. One is the monoclonal antibodies, which are the specific antibodies which actually occupies this ligand binding site and prevent this homo and heterodimerization. However, the other one is the tyrosine kinase inhibitor, which are actually basically a small molecule inhibitors. They occupies this ATP binding site in the kinase domain and prevents this autophosphorylation and abrupting this cancer cell proliferation. And in the last 20 years, there has been lots of tyrosine kinase inhibitor discovery has been made in this EGFR to combat the EGFR mediated cancer. The first EGFR tyrosine kinase inhibitor was the gefitinib, which got the FDA approval in the year of 2003. But due to the emergence of T790M mutation, where threonine 790 amino acid got mutated with the methionine, and it causes the resistance against the gefitinib. And gefitinib was in reversible nature. So after a certain period of time, it was coming out from this ATP binding pocket and causing this again onset of the cancer. So to overcome this problem, researchers came up with the efatinib and canatinib. These two drugs were like categorized into the second generation of EGFR tyrosine kinase inhibitors. They had this Michael acceptor group, which were actually forming a covalent bond with the cysteine amino acid residue present in the kinase domain. But due to the off-target affinity towards the wild-type EGFR, these drugs were also considered as a drug resistance category. To overcome from these two problems, third generation of EGFR tyrosine kinase inhibitors were developed. Among them, osimertinib was developed in the year of 2015, and osimertinib was very potent to combat the resistance issue in this T790M mutant form of the EGFR. But in the year of the same year where osimertinib was discovered, in that year, C790S mutation was discovered, which causes this osimertinib was also drug resistance. So these are the problems. So taking these problems, we hypothesize what can be the next approach to combat this continuous emergence of the drug resistance. And naturally, natural compounds are like bedrock in the drug discovery, and among them, Taxol, penicillin, statins are already in the market. However, several natural compounds such as EGCG, thymoquinone, vincristine are well known plant-derived ethnic acid drug. And why natural compounds? Because they have less prone to develop further drug resistance, and they have less cytotoxicity towards the normal cell, and they have greater molecular rigidity compared with the synthetic compound, and naturally they have higher number of the hydrogen bond and donors and acceptors. So the major question we had, what natural compounds shall we select for to develop a EGFR tyrosine kinase inhibitors? So we came up with the green tea, which green tea is the second most largely consumed health beverage across the world. And among them, the major health beneficial property of the green tea comes from this compound called EGCG. It's a epigallocatechin 3-guanidate. It has 8 hydroxyl group, and it's a very powerful antioxidant compound. But when we did further research, and we found that EGCG has several health beneficial property ranging from antioxidant, anti-cancer, anti-inflammatory, antiviral, etc. And EGCG have already shown a to inhibit the growth of osteosarcoma in this in vivo model. But then we had a question: if EGCG has this much of promising wide range of health beneficial activity, then why they are not being used as a anti-cancer drug? Then we found that they have certain drawbacks, which includes chemical instability, low bioavailability, poor membrane permeability, and unknown mechanism of action. So then we came up with that can structural derivatization of EGCG can lead to the superior compound. So with this, we prepared a synthetic route to synthesize 4 prime alkyl derivative. Why this particular position? Because in our lab we found that this particular 4 prime alkyl position is cannot hamper the original property of the EGCG. So we wanted to take this advantage. So we came up with alkyl bromide at various chain length and in the presence of sodium anhydrous acetate and dimethyl formamide. We carried out the reaction for it at 85 degrees Celsius for 2 hours and we got the product, where N denotes the 6, 8, 10, 12, 14, 16, and 18 carbon chain length. And we did this synthesis with the ether bond, and ether bond will prevent the further hydrolysis of this particular group. The previous efforts has been already made to synthesize alkyl EGCG derivative, but those were with the ether bond and ester bond, and ester bonds causes the esterification in the body and they further gets hydrolyzed very easily. So to confirm our synthesis, we first purified these derivatives and we carried out the HPLC, and we in the HPLC analysis we found that these compounds are more than 98% pure. And for the structural characterization, to confirm that indeed this structural derivatization has happened at 4 prime position, so we carried out 2D NMR where we confirmed that indeed this structural derivatization has happened at the 4 prime position. So we taken 3 cancer cell lines. These 3 cancer cell lines were having a wide range of EGFR expression, where in A431 cell line, it's the highest endogenous EGFR expressing cell line. However, HeLa expresses moderate level of EGFR and MCF7 expresses low level of EGFR. And we also taken HEK293 as a non-cancer cell line to see that is there any effect of these drugs on the non-cancer cell line. And we carried out cell viability analysis by using CCK8 SA-KIT, and we found that one derivative named 4 prime C14 showed the promising anti-cancer activity against these 3 cell lines. As A431 expresses highest endogenous EGFR expression, which was confirmed by Western blot and immunofluorescent analysis, we taken this cell line for further investigations. And when we did the EGF stimulation in the A431 cell line, we found that 50 and 75 macromolar of 4 alpine C14 EGCG was able to inhibit the phosphorylation of the EGFR. In this experiment, we also taken gefitinib as a positive control, and we carried out the immunocytochemistry analysis, and wherein we also confirmed that 75 macromolar of 4 C14 EGCG was indeed able to block the phosphorylation of the EGFR. These immunocytochemistry analysis was in line with our Western blot investigations. So next, we wanted to see that effect on the downstream signaling cascade. So we've taken 2 important protein, phospho-ERK and phospho-AKT, and as we can see from immunoblot analysis that blockade of EGFR resulted in the downregulation of these 2 downstream signaling cascade, and further they induced the cell death. To identify the mediation of the cell death in this A431 cell line, we found that BCLXL, which is an anti-apoptotic protein, was downregulated. However, BAX, which is a pro-apoptotic protein, was upregulated. So this A431 cell death was mediated through the induction of intrinsic apoptosis. Our EGCG is well known for its antioxidant capability. And then we carried out DPPH assay, where we found that there was not significant difference between EGCG and 4-C14 EGCG in its antioxidant capability, suggesting that introduction of 4-alkyl chain at 4 prime position doesn't hamper the actual property of the EGCG. However, as I said in my introduction slide, that EGCG has a drawback of poor stability. So we carried out stability analysis at 5 hours and 24 hours, and as we can see that 4-doliprane EGCG, after 24 hours, there was only 27.80% degradation happened. However, EGCG was 50% degraded in this PBS, which resembles of our physiological pH, suggesting that introduction of alkyl chain also improved its stability of the EGCG. So recently we did literature search, and we found that cytoprotective autophagy has been emerged as a drug resistance mechanism, where if drugs such as tyrosine kinase inhibitors or other inhibitors are introduced to block the receptors, so these pathways are getting downregulated, but there are genes which is autophagic genes, they are found to be upregulated and they prevent the cell death, so causing these tyrosine kinase inhibitors the drug resistance. So we come up with a strategy that if we synergize anti-cancer drug along with autophagy modulators, we can enhance the therapeutic benefit to kill the cancer cells. So we taken here glioblastoma cell line. For this analysis, first we carried out flow cytometry analysis to check that cell death of 4-doliprane 10 EGCG and other derivatives of the EGCG, and we found that 4-doliprane C12 and 4-doliprane C14 EGCG induced 42 and 36.89% of cell death respectively in the glioblastoma cell line, and we found that C12, C14 EGCG were able to block the phosphorylation of the EGFR as well as downstream signaling cascades. When we checked the autophagic marker, which was LC3B1 to LC3B2 conversion, we found that those derivatives which block the EGFR were also able to induce the LC3B1 to LC3B2 conversion. And we also found that sequestrome protein was downregulated, suggesting that these drugs are, after blocking the EGFR, are inducing the cytoprotective autophagic response. So when we combine the our EGCG derivatives along with the chloroquine, which is a late autophagy inhibitor, we found that enhanced cell death in the glioblastoma cell line, where we found that alkyne EGCG along with the chloroquine 20 micromolar induced the maximum cell death in this glioblastoma cell line, so giving us a new approach to tackle the drug resistance. So in summary, we taken an alkyl EGCG derivative which blocks the EGFR and downstream signaling cascade, but this blockade indeed activates the cytoprotective autophagy. But when we combine the chloroquine, which blocks the autophagy, and our alkyl EGCG derivative, which blocks the EGFR protein, certainly leads the apoptosis, which resulted in the enhanced cancer cell death. So one more thing here we wanted to explore that effect of these derivatives on the mutant form of EGFR. So as T790M mutation, which comprises the 50% of the drug resistance amongst other mutation, so we carried out the in vitro kinase assay where we taken gefitinib, which is a first generation inhibitor, WZ4002, which is a third generation inhibitor, and we found that 4-doliprane C12 and C14 EGCG were mutant selective as they blocked the kinase activity at 18.39 and 33.33 nanomolar. So we wanted to explore that how these compounds actually binds with the interact with the EGFR protein. So we carried out the MD simulation analysis for 400 nanoseconds, and we found that these derivatives were very stable throughout the MD simulation, and they resulted in strong binding in the EGFR mutant EGFR binding cavity, which resulted the maximum binding for 4-doliprane C14 EGCG followed by C12, C10, and then parent molecule EGCG. So coming to the conclusion, so here in my PhD work, we have developed a potential third generation EGFR tyrosine kinase inhibitor from the natural product origin, and they were capable in blocking the double mutant EGFR, and they have increased binding affinity toward the mutant EGFR as confirmed with MD simulation analysis. And coming to the drawbacks of the EGCG, which was the poor stability, as we found that improved stability compared to parent molecule EGCG, and antioxidant property of 4-doliprane C14 EGCG was also not significantly different from the EGCG, and they selectively target the cancer cell, sparing the non-cancer cells. And as cytoprotective autophagy is the one of the major cause for the drug resistance, and we found that if we combine the autophagy inhibitor along with our natural compound-based EGFR inhibitor, they were able to enhance the cell death in the glioblastoma cell line. However, in our future approach, we are rationalizing and designing the project where we will evaluate the in vivo pharmacokinetic and thermodynamic study to assess the efficacy of these alkyl derivatives. And also, we are curious to see the effect of these derivatives against the third generation mutation, which is a C790S mutation and exon 20 insertion mutations. I would like to acknowledge my thesis supervisors, Professor Avinash Sonawne and Dr. Sushaban Sadhu Khan, Department of Bioscience and Biomedical Engineering, IIT Indore, IIT Palakkad, Ministry of Education, Team India Bioscience, then my collaborator, Dr. Parmalkar, for MD simulation analysis, my lab mates, and thank you. I'm open for the questions.
Thank you, Satyam. Excellent talk, and you finished well in time. So well done. That's your opportunity to
left.
Great. Thank you, Manjula. Thank you. 4 minutes left, says Manjula. It's an opportunity to get feedback from the expert mentors. So expert mentors, Kiran, Aarti, and Ram,
Can I
please share your
start,
thoughts.
Krishna? Yeah. So maybe some small questions I have for Satyam is that when you do the alkylation and derivatize this molecule, because we had two concerns, bioavailability as well as the membrane permeability.
Yes.
So which one do you improve with that alkylation?
Sir, can you repeat one more question? I missed the last part.
So I'm saying that with the derivatization with alkyl group attachment,
Yes.
You improve one property of membrane permeability or the bioavailability?
Actually, we checked that mainly. Actually, the EGCG has three major concerns. One is the stability, then bioavailability, and then poor membrane permeability. So initially, insofar I have studied for the stability of the EGCG, and we checked in the in vitro assay. We haven't done any experiment in vivo. So in vitro assay, we found that like poor alkylation of the EGCG has resulted in the improvement of the stability of EGCG. So we haven't done any bioavailability assay or membrane permeability.
Because some of the later experiments, you apparently see that C12 derivative is probably behaving better, particularly in the autophagy assay, right? So
Yeah.
any reason to choose C14? Because other parameters were also in the similar range for C12 versus C14.
Yeah, actually, when actually the C14 was very consistent through. Actually, we taken multiple cell lines to shortlist this derivative. So as we from initial study, when we checked in high EGFR expressing cell line to low and moderate level, so C14 was the very consistent one among the compared with C12 also. So, but the ability to inhibit the EGFR was like both C12 and C14 were very comparable. However, C12 EGCG and C14, if you compare both, then C14 was so far in my study has shown like better efficacy than C12 EGCG.
Okay, and maybe one last question or suggestion I have is that when you are using glioblastoma, do you know if you're targeting through EGFR only or some other tyrosine kinase receptor is working there?
Yeah, so actually for that reason, actually we checked the first expression of the EGFR protein in the glioblastoma cell line. So we found that there was like significant overexpression of EGFR protein was there in the glioblastoma. To see that actually we got this question before also. So that's why we carried out the in vitro kinase assay where we taken the pure EGFR protein to see that like how much is specific of this drug to block this EGFR kinase. So we found that these derivatives were very selective as they shown in nanomolar range to inhibit the kinase activity of the EGFR. However, we are also planning to screen like all other kinases in the in vitro assays so that like we can have a better idea like these drugs are actually targeting the EGFR protein, not like other drug targets.
And do you know if your EGCG is specific to EGFR? Because particularly the inhibitors which are targeting the tyrosine kinase domain, they have very broad spectrum. Basically, it's not just for EGFR, it could be any other tyrosine.
Yes, for that only reason, actually, we selected that A431 cell line, sir, which has like highest endogenous EGFR expression. But you are very right that like because these are the natural compounds, so they have broad spectrum of the drug targets. But to emit that possibility, we taken the highest endogenous EGFR expressing cell line. However, our plan is also that, so that's why like we wanted to test that in vitro, that like these drugs we wanted to pull down actually. So in our lab we are also have designed alkyl EGCG, which has alkyl group, so which will provide us a ability to do the click reaction in the in vitro condition, so that we can pull down the targets of the this alkyl EGCG in the in vitro system, and from the mass spectrometry analysis we can confirm that like what are all the actual targets of the EGCG inside the cellular environment. So we have already designed the alkyl EGCG, and we have recently reported, other members from my lab actually they have reported that these alkyl EGCG indeed are targeting this kinase receptor molecule, but indeed we are yet to confirm that they specifically targets the EGFR or not.
Okay. Maybe one last question I can ask Karishma, right? Or maybe suggestions, right? So
Yes, we have time.
yeah, because you said that EGCG to begin with, it's enriched or it is there in the green tea, right?
Yes.
And the concentration which you are using. So what kind of relation is there that from green tea, how much do you get, and what kind of concentration you are using in these inhibition assays?
So we didn't directly isolate it from the green tea actually. So we taken the purified compound of EGCG first. So we purchased directly from purified compound EGCG, and then we did the synthesis. So initially we started the activity of these derivatives from 10 micromolar to 75 micromolar. But from throughout my study, we can see that these activity of these compounds in the in vitro condition, in the cellular condition, they are actually giving a good activity at from 40 micromolar to 50 micromolar range.
Okay, yeah.
Thank you, sir.
Thanks, Ram. Thanks, Satyam. Aarti, Kiran?
Yeah, sure. Dr. Ram has covered a lot of points
Okay.
already. So I will just add to that. I mean, just a general comment about the presentation itself. Like, I felt like there were tables, there was a tables overload. So you might want to consider like simplifying some of that data so that we can keep track of it. But can you, to come back to one of the points that was brought up, where the membrane permeability of these alkylated derivatives now, what would you predict? Because you haven't tested that. So I'm a little concerned that the stability that was tested in vitro, how would you relate that to the final bioavailability, which will also be a function of membrane permeability of these molecules? Like, don't you think that is important to test?
Yes, ma'am. Indeed, it's a very great question actually. So there was like when I was doing this work, there was at the same time there was a literature actually published. So they also synthesized certain more or less similar kind of derivative, but they did the ester modification. So they here in my compound we have did this ether modification, but they made this alkyl addition by using ester modification, and they also modified this B ring also. So then they did like this permeabilization assay, and as the this hydrophobic molecule, so there is a lipophilic interaction happened, and they reported that like introduction of these chains are actually improving the membrane permeability of the EGCG by introducing the alkyl chain. But as like in our laboratory, we haven't carried out this experiment, as we are first that's why we before starting the xenograft experiment. So we are starting with the pharmacokinetic experiments first, because pharmacokinetic analysis will actually give us the actual idea like at what time till what time our drug is actually present in the what is the actual plasmatic concentration of these drugs in the in vivo condition, and then there we can figure it out like what can be done to improve, either we have to improve it or we can go with these compounds only.
So another big question I had was that what is the mechanism to, from what I understood, by the time you came to the autophagy part of this towards the end, you were additionally using an inhibitor for autophagy, right?
Yes.
So the claim that this molecule has the potential to overcome cytoprotective autophagy seems a little stretched to me. But wasn't that because you added an additional inhibitor?
Ma'am, actually, there is actually two types of autophagy. One is the cytotoxic autophagy, which actually favors the cancer drugs. But when this cytoprotective autophagy comes up, actually what happens when you give a drug to the cells? So autophagy is like to prevent the cell death actually. So the cell actually thinks that these drugs are a foreign molecule, so they eventually protect the cancer cell from killing it. So what is happening actually when we were blocking the EGFR compound? So in the downstream signaling, there are like this autophagic protein were actually getting upregulated. So when we are blocking the EGFR protein, but in the meantime, this autophagic response is getting triggered, and they are protecting the cell death. But cell death indeed cell death is occurring. So from the cell viability analysis, if you see. Alone, photoprime C14 EGCG were able to kill 50% of the cell death. But when actually we are blocking the autophagy by using this chloroquine, so it is actually enhancing the cell death. So this means like because this gefitinib which is a promising EGFR inhibitor, but the problem with gefitinib is actually continuous emergence of multiple drug resistance, and cytoprotective autophagy is one of them. So that's why like alone one drug cannot like what we predicted, alone one drug cannot uh, uh, overcome the drug resistance. So that's why like we thought to use this chloroquine to block the autophagy at one stage, and the EGFR is blocked at other stage. So this can further enhance the therapeutic efficacy of any compound. It's not like here we wanted to give us an idea that like this strategy can be utilized with other compounds also, which is falling under the drug resistance category.
Okay. Okay. Thank you.
Thank you,
Thank
Aarti.
you.
Thank you, Kiran.
Yeah.
We can. Yeah.
Yeah, so I had a similar question as Aarti on this autophagy, but more fundamentally, you said you don't know how the target for your modified EGCG, right? You still don't know the target. If I remember correctly, you said
Yeah,
yes.
so like whatever we have done so far in vitro experiment, so as we don't have done any like experiment which actually directly confirm that these drug indeed only targeting the EGFR and not targeting other compounds, other targets actually.
Okay.
Because it's like natural compounds, so natural compound have tendency to target others proteins also.
So because in this diagram that you are showing right now, you have selected some double mutant of EGFR, right?
Yes, yes.
Is that drug resistance evolved under some selection pressure?
Yes, yes. So
So is this specific to that particular mutant, or is it a bit more promiscuous in its activity?
So what happening actually? So EGFR has like certain like there is like lots of mutations in EGFR. So initially we wanted to first make sure that these compounds are very they are able to block the EGFR or not. So we tested then we found that like T790M mutation, which is a first generation mutation, and L858R. So these mutations are actually causing the 50% resistance of the available tyrosine kinase inhibitors. So that's why this make us very curious to see the efficacy of these derivatives against this particular type of mutation. So we found that indeed they were selective compared to the wild type toward this mutant, but as recently like C790S mutation, which is actually causing the rest 25 of drug resistance, we were also curious to see that like how these derivatives behaves against that particular mutation of the EGFR.
Okay. So I have one or two very probably naive questions because I don't work on cancer. So this overexpression of EGFR, is it a cause or consequence?
It's a cause actually. So so far researchers haven't identified how this EGFR gets upregulated, but like there are certain that common cause that initially they predicted that those smokes a lot they get upregulation or overexpression of the EGFR, because EGFR is very crucial for the normal cell proliferation. So, which makes it very obvious to say that this particular protein can be overexpressed when any kind of cancer occurs. But in the mainly in lung cancer, there is a like 50% of the chances that EGFR mutation is happening to cause this lung cancer.
So if EGFR is overexpressed in normal cells, it will not cause cancer, right? Or does it?
So the like if it's overexpressed, then it will cause the cancer. If it's like normal, like routinely activated, because it's a signaling protein. So in our body, we continuously need the cell cycle gets activated, then stop, then activated, then stop. But when overexpression occurs, then this cell cycle continuously goes on, and it causes the cancer actually. So that's why overexpression is harmful for our normal cells. But if it's like in controlled way, then it's very important for normal cell proliferation.
Okay.
Okay.
Yeah. So
Yeah.
my last suggestion would be like as Aarti suggested, slides are a bit overloaded, and maybe you can just use less data per
Slide.
slide. And
Yes.
maybe you
Yes.
can also speak a bit slower so that we can follow you. Yeah. And those are
Thank
my
you, sir. Thank you for suggestion. I'll
thank you.
keep that in.
Yeah, thank you.
Thank you, Satyam. Good talk. Satyam, some of your colleagues have had some questions in the chat window. Take a look there. Sweetie, you're next. As you put your video audio on and start your slides. Um, Go ahead, Sweetie. Satyam, you can stop sharing. So I think Kiran, Aarti, and Ram, we told our PhD speakers 14 slides. They found a hack around it by adding a lot of data onto the slides, which is natural. We've had that through the session. Everyone wants to talk about everything that they are doing. So totally, totally get that. Well done, Satyam and Sweetie. You're next. Yeah, we see your slides, Sweetie. Go
Yeah.
ahead. Your
Okay,
20
cool.
minutes
Yeah.
starts now, Sweetie. 20
Yeah.
minutes
Am I
starts now.
audible?
Yes.
Yeah. Okay. Great. So, thanks to all the organizers for this opportunity. I am Sweetie from Dimple Notani's lab at National Center for Biological Sciences, TI for Bangalore. So, I am a final year PhD student, and I'll be talking about my PhD work again. So, we have looked at a histone modification and how it can play a role in preserving interface chromatin memory. And I'll come to what I mean by this memory here. So we have all seen this schematic at one or the other time in our studies. So this is a diagram by Fleming where he observed, and this was like late, uh, long back in 1800s, where he observed at the cells in a basic light microscope, and he saw that the cells were dividing. What he interestingly saw is that the chromatin, which was like this, so chromatin is essentially the DNA which is packed inside the nucleus. When the cells were dividing, that is in different phases, the DNA will take these different shapes. So from something like this, it goes to rod shaped structures, which are chromosomes, which we essentially call chromosomes. So the idea was that when this is happening, now after so many studies, we know that there is a drastic loss in not just in the structure but also the function of chromatin when the cell divides. And we know that multicellular organisms have to divide. So the key catch here is that the structure that I was speaking about gives the cell type specificity in the multicellular organisms, and this is only during the interphase. So if I go back here, these are interphase cells which will have different gap phases or S phase where the DNA is replicating. After this, the cell enters the M phase where the chromosomes have to divide and make two copies, and this is essential for normal growth and development. So I'll just spend some time here because most of my work is around this. So from what studies have shown till date is that DNA is not naked, of course. It is wrapped around histones to keep it compact. And these histones, they form these optomers called nucleosomes, which can occupy different positions. And the modifications that happen on these histones essentially can make the region active or repressed. So when I say active, the gene can be transcribing in a given cell type, and if it is repressed, meaning the DNA is more compact and not accessible to transcription factors, it will be not transcribing. And there is additional regulation that happens on this, which is transcription factor binding and chromatin remodeler binding. And what they do is that suppose if you have a gene here with a promoter, which is a cis regulatory element, and then an enhancer element, which is essentially another DNA element on the same long stretch of chromatin, which has to come close to its gene because both of them have different machineries which come together and now can activate the gene transcription. So you will notice that there are these like lots of loops of DNA in the interphase nucleus. And these loops are formed by the different enhancers and promoters coming together. Now, this is essential because if a cell has to respond to some external stimuli, these loops can be dynamic. The genes can come closer to their enhancers and maybe move apart when not required. But these loops do not, they occur in these entities called topologically associating domains, TADs. So what TADs we can think of is if a thread is DNA and if you start making knots at different places, then these knots will be different TADs. And these are important because a given set of genes which are to be, let's say, regulated together because for if it's a heat shock or if it's a salt shock, they stay together so that whatever transcription machinery is required is in close proximity and can be activated when required. So these are TADs. I'll be using this term very often. So these are TADs or domains. These TADs or domains now, as I said, for a given cell type, the gene can either be active or repressed. So if it's active, it falls in an active domain. So it will be part of one of these TADs and it will be part of a domain, which is active domain. And then the genes which are not active will be sort of clustered together in these different TADs, forming the B domain. Now, in an interphase nucleus, when the nuclear lamina is present, the chromosomes are present in chromosome territory. So they occupy distinct spatial spaces. And within them, there are these domains. So all the DNA in a chromosome which is active, it's part of an active domain, and the part DNA which is repressed is in the B domain. Now, these domains will vary from each cell type to another cell type. Now, the interesting thing is when the cell is dividing, all this structure that I just spoke about is going to take a different shape and you will not see any of this hierarchy because now the DNA is condensed into these rod shaped structures which are chromosomes which you have all seen. So the question that people started asking about like almost 100 years after Fleming saw those like cells was that how is the cell remembering this identity, because essentially in mitosis all the cells are more or less alike because you're losing the structure, and not just this, you lose a lot of modifications of histones, you lose transcription factors. So then this concept emerged which is called mitotic bookmarking, where if you assume that a cell is this book and you want to bookmark different pages, so each page is a gene, and for a given cell type or a given book different pages are important, so you will bookmark those by keeping certain transcription factors or histone modifications. And so now once the revision is over, which is this stage, after this when the cell is coming back to the same interface, the cell will remember, okay, this gene has to be activated. But the question that is not yet addressed, or in it's very preliminary, is how is the domain memory retained, because here I spoke about bookmarking which is more local bookkeeping of the genes. But what about this big structures, the TAD structures, which are like megabases of DNA? How do those structures come back? And when I say come back, I would want to spend some time again on this schematic. So this data is from HiC, which is where you want to see if one piece of DNA is interacting with the other. You do fix the cells. Now, whatever DNA is sticking together through some downstream processing, you will capture those interactions. Now, the diagonal that you see here is this is part of chromosome 21, meaning that each DNA element is going to interact more with its neighboring element, which is intuitive. But additional to that, you see some stripes and dots here. These are indications of the TADs that I just spoke about. And the same can be seen in this line plot. So this is TAD signal, this is compartment or the domain signal. You see alternating domains. So an active domain, then a repress, then again an active repress, and so on. And different TADs. So if you focus on these lines from the gap phase to the DNA replicating phase and then to the M phase, you see the lines get flatter and you don't see any of these interactions because these structures are lost in the chromosomes, the rod-shaped chromosomes that we just saw. But it is coming back again after the mitosis stage in the next interphase. So we ask this question, can a histone modification, which is abundant in mitosis, take over this function and remember where the domain was? So what we looked at is a histone phosphorylation. So just to give a brief overview of how each modification looks. So this is actually a bit off here. So histone acetylation decreases, histone methylation is consistent. Histone phosphorylation was supposed to be maximum here, but what I mean by maximum is, so if you see the schematic, from different G1s to G2 phases, and then in the M phase you see this red color on the chromosomes. This is the histone H3 S10P. So the histone 3 at serine 10th position is getting phosphorylated. So it's abundant in mitosis and it's pretty conserved. So from tetrahymena, which is a protist, to mammalian cells like HeLa, MCF7, different cancer cells, and even stem cells, you see that the condensed chromosomes have H3 S10P or the serine 10 phosphorylation on histones. So we then ask this basic question, that the H3 S10P which is present on these dividing chromosomes, can it retain the structural and functional memory of interphase chromatin? So once the cell is back again to the stage, can it retain the identity? And for this we are using HeLa cells, which is a continuously growing culture. So you will see most cells in interphase, only a few cells are in dividing phases. So what we do is we enrich the mitotic population in HeLa cells by giving nocorozole treatment, which arrests the microtubules and you don't let the chromosomes segregate, but they are condensed. So then we do a experiment which is called chip sequencing. So again, chip sequences is when you want to see on your DNA what protein is binding. So here you again fix the cells and then pull the DNA with specific antibody. So here we are using antibody for H3 S10P, and you will enrich the DNA regions which will have this modification along the DNA. So what we see here is that you do some downstream analysis which tells you that from asynchronous, which is all the population of HeLa, versus the cells which were enriched in mitosis, there is a fourfold increase in the regions which have the modification. And when we ask what these regions are, so you can always go back to the genome, and we already know which regions are promoters, which regions are enhancers. What we notice is that from asynchronous to mitotic stage, you have an increase in the promoter regions. So promoters are the positions where like various transcription machinery is going to bind, and the transcription is going to start. So it's an essential gene regulatory region. And the same, like how the chip-seq signal actually looks like, is this. So you have enrichment and then an under enrichment. And if you compare this with another histone mark, so H3K27 acetylation. So acetylation is essentially it makes the DNA more accessible. So it's a very standard enhancer promoter mark, the acetylation mark. And if you overlap that with this, you see the enhancer doesn't have it, but for a MYC gene where the gene is starting, you have the modification for H3 S10P. So essentially, from this, we can conclude that the H3 S10P, which is increasing on mitosis on the chromosomes, is enriching at promoters. But more interesting than this is that if you keep zooming out on the DNA, this is, I'm showing whole of the chromosome 11 here, then you will see that the H3 S10P is actually forming these domains. So if you follow the line that I am doing, so it's over enriched and then under enriched and then again there is enrichment. And if you go back and overlap this with the A and B domains, so remember this A and B domains are active domain and repressed domains. Again, positive axis shows A domain, the negative shows repressed domain. And this data is from interphase because in mitosis these domains are absent. So if you see in interphase wherever an A domain was, in mitosis H3 S10P is increasing there. And this is quantified here in this profile plot where you can see from blue to red, which is from asynchronous to mitotic condition. On A domains H3 S10P is increasing. So from here we can say that in mitosis where the A and B domain or the TADs are absent, H3 S10P is actually present along the DNA on those domains. So there's nothing stopping H3 S10P from to spread here, but even then it doesn't. So that means it's giving some memory to the cell for the domains. Then we ask, is this all the domains are equally, equally having the modification, or is there a difference in terms of its function? So then we divide these domains into three categories based on the level of H3S10P in mitosis. You see an similar trend for acetylation. Like I said, acetylation is an active mark for the interface cells. So you will see the same trend, which means that H3S10P is not only marking these domains along the length, it is actually present more at the more active domain and less at the less domain. So it's kind of a rheostat for the activity in terms of its deposition. And this is another plot which is a more like so the TADs which I just mentioned, when I say the genes which are present in TADs, they have to be regulated together. They are better regulated together if their boundaries are more insulated. So if there are many genes here and the boundary is insulated better, then what happens is that the regulation is more strictly controlled. And that is something that we test with another protein binding which is called CTCF. So these are two boundaries of the DNA and there is a lot of DNA inside it. And what we are seeing here, again for the TADs which have higher SNP versus low mitotic SNP, the boundary insulation is better because the CTCF is more present at the TADs with higher SNP. So from this again we conclude that H3 SNP is not only marking these but also it's giving some idea of what the activity may have been in that domain. Now, H3S10P has been associated with chromosome condensation in mitosis. So, if we ask if the H3S10P that is present in mitosis in these domains, does it have anything to do with the compaction? And indeed, if you see this heat map here, then this is the TAD with high SNP, low SNP, and then again a random TAD control. So, you see more blue color here means more interactions. So, what we are seeing here is that there is more local interaction in the TAD where SNP is present. So what SNP is probably doing is, if you have a domain which is active in interface, it gets loaded on, the phosphorylation gets loaded on the serine residues of those histones, and those histones then reel in the DNA such that the region becomes more compact. So physically it is giving its identity, and then functionally also once the phosphorylation is removed, it can give the meaning to it in the next interface. Now the question was, if in mammalian cells, the HeLa cells, if we have the mutation, the phospho dead mutation and the phosphomimetic mutation, which is alanine, serine to alanine and serine to aspartate, will it have any effect on the gene expression? Because that was the whole point of the study from the beginning. So then we made these three lines in HeLa cells, which were wild type, phosphodid, and phosphomimetic. The good part about this is that these are inducible cell lines. So once we give doxycycline, only then the cells express the HA tagged histones, which can either be the two mutants or the wild type. Otherwise, they don't express. And this we used because we want to test the absence of serine 10 phosphorylation only before the mitotic and the interface that we want to collect the cells in. So we are doing this induction just before the stage where we want to study how it affects the chromatin. Sorry. So I'll just quickly go to what we did in this experiment. So in mitosis, when we harvested cells, after giving the cells either a wild type or the two mutants, what we notice is that in mitosis, if you look at these chromosomes, so these are the chromosomes from the three different cell lines, and the experiment is metaphase spreading, where you just swell the cell and then drop it off from a height such that now their chromosomes are spread out and you can look at their features. So we are looking at their compaction right now. You see that the two mutants, they are now less compacted. And this is probably because the serine 10 is not getting, is not there. It's alanine or the aspartate, which are not getting phosphorylated. Therefore, you don't have the compaction that is required. Then we looked at the um, next interface transcription. So I'm not showing a lot of data here, but we tested this compaction defect at various scales. And we then tested that in the next interface, so which is this stage. After inducing the system with doxycycline and inducing these mutants, in the next mitosis, which is where the identity rule will actually have an effect on, we asked if the transcription is getting affected. So to be frank, we were not expecting very drastic differences here, but we still noticed subtle but yet robust differences. So we have put some strict thresholds here, and what you notice is, if I take the transcription in HeLa cells in this order, so these are the genes which are arranged in increasing order of transcription, you notice that for both the mutants, which is the alanine as well as the phosphodiesterase mutant, both of them are showing us that for the genes which are more active, they tend to express more in the next interface. So what we are proposing with this is that H3SNP, which is a serine 10 residue of histone 3, when it gets phosphorylated, it essentially happens on the more active domains, and such that these active domains are now more compacted in the physical space. And this data that I have not shown is that these domains take less time for reformation in the next interface and are more active in the next interface. But once you express the mutants, what happens is that this compaction that was happening with the help of H3SNP is not allowed to happen anymore, because the nucleosomes become unstable, the mutant nucleosomes, and the DNA is almost like everywhere. So the genes which were closed, they are now not closed, and since they were already active, they get even further activated. So the um, the amount of activity that a promoter, the gene promoter, should carry in a mitosis stage was controlled by H3 phosphorylation, but since it's not present anymore, the promoter now gets additional component of expression in the next interface. So this would mean that, suppose if you were to express these mutants for very long or continuously throughout the stage, we expect that maybe the cells which are expressing a gene, it will be a system where you will only see expression of certain genes, and the genes which are downregulated will just die down. So you will get a very drastic difference in terms of gene regulation rather than the grade of gene regulation that you see. So with that, I would like to acknowledge my thesis supervisor Dimple, my TCA members, Raj from NCBS, Ravi and Sachin from ISC, and Sachin, who helped me with a lot of analysis experiments, and then other people from Dimple Lab and the other authors in this manuscript. Special thanks to SIF and NGS at NCBS, which are the flow cytometry and the genome sequencing facilities. And then Shrimonta and Avinchal from ISC for help with some MESC cultures, different funding sources. And yeah, if you would like to read more, maybe you can look at the preprint, and I would be happy to take questions.
Thank
Thank
you,
you.
sweetie. Nice talk. Aarti, Ram, and Kiran, we have time for questions till 11:08. 10 minutes.
Can I take
Sounds good.
a couple of quick questions?
Sure, sure.
Yeah.
And sweetie, the more questions you want, the quicker and on point you can keep your answers.
Yeah.
Go
So,
ahead.
Sweetie,
Yeah.
nice presentation. Couple of questions I have. First thing, it is already known that what is the kinase or phosphatase which is working on the phosphorylation, dephosphorylation of the histone 3 is serine 10?
It is known. So the aurora kinase B, which is a cell cycle kinase,
Okay.
it is
Okay.
the one which majorly phosphorylate, but then there are a couple of other kinases, for example, aurora kinase A. And then in
Okay.
interphase also, it gets phosphorylated, MAP kinase, CDK, etc. But mitosis majorly it's aurora kinase B.
Yeah.
For the
Yeah.
phosphatase, it's PP1C subunit. So the big
Okay.
phosphorylation dephosphatase unit does that. Yeah.
So is there any proximity thing which drives this, meaning the kinases or phosphatases to the chromatin region?
Right.
Active, because you said
So,
that this is happening more on the active chromatin region, right?
right. Yeah. So, only one study is there, like a rigorous study, where they show that when the deacetylases are removing the acetylation, they bring in this aurora kinase to the DNA. So, wherever there was high acetylation, the deacetylase will sit and remove it, and the aurora kinase B will be attracted to those locations, and now it will phosphorylate the residues. So there is one study that indicates it can be mediated that way, but yeah,
Okay.
only that.
And one thing I want to know is like, so you're using HeLa cells for this study, right?
Right.
Do you
Yeah.
think the scene will be very different if you use actually a diploid cell line like DLD?
Yeah, we actually had given a thought about it. So, but of course, it will be a very good idea to use a deployed cell line. Only problem will be that you will have to generate a lot of these interaction data sets and the mitotic release data. So I didn't show a lot of data here, but in HeLa cells, people have looked at the chromatin interactions during mitosis, after mitosis, etc. So the picture may or may not remain the same, but yeah, I mean, I can't really comment on whether the deployed will be any more different in terms of results, but it's not a good idea at this point because of the limitation of data sets. Yeah.
And maybe last question from my side is that when you make the mutation, whether it is a phospho-dead or phosphomimetic one, right?
Uh,
Why do you see the change in the same direction? I would expect that they should be on
yeah,
two sides as compared to the control.
right. Just give me a minute. My laptop is on low battery. I'll
Yeah,
connect with my phone and then answer this.
yeah.
Just a second. Yeah. So sorry. Um, but yeah, while the laptop is still on, so actually it turns out that this modification, when it's getting phosphorylated, the act of phosphorylation and the act of dephosphorylation is crucial. So whether you carry the charge or not is probably not important. For chromatin, what it really cares about is whether the protein machinery is coming for the phosphorylation or the dephosphorylation. If they don't reach in time, then probably you will not see the effect. That's our understanding for now, but no experiments to really see that. Yeah.
Okay, thanks.
Okay.
I just
I
had a
will
few.
connect with my phone. Just give me a minute.
Yeah.
Yeah, should I go ahead?
Sure, Karan, please.
Yeah, great talk, sweetie. Nice work. So I just want to know, again, my understanding is very limited on this. See, what is the half-life of this phosphorylation of this H3S10? Is anything known?
Right.
Sweetie, the question was, what is the half-life of the
Yeah,
phosphorylation
yeah.
of
Yeah,
this of this H3S10P?
right. Sorry, I really have not a clear idea on that. But what I know is that this phosphorylation, so if we consider that M phase is 20 minutes and the initial period is for prophase, prometaphase, when it's more abundant, is let's say 10 minutes for HeLa cells. Till then, the phosphorylation is seen on the chromosomes, but it's very dynamic. You will see, if you stay
Yeah.
in the cells, you will see it like at different locations. So probably half-life is playing a role, but I am not sure if the same residue sticks on or yeah. I mean, chemically, I really don't have an answer.
So it is highly dynamic, and also probably it is context dependent as well, right?
Right.
Yeah. So yeah. So the other question I had is, you have also done this S10 mutation.
Mm-hmm.
Is there any redundancy in terms of how the compaction is maintained? Because it is highly unlikely that our cell system would depend on phosphorylation of a single residue, right? It would not bank on
Right.
just
You are
one. Yeah.
right. You are absolutely right. So there are, in my knowledge, there are like three different modes. So if we talk about histones, it's the acetylation of histones, it's the methylation as well as the phosphorylation. So methylation is as consistent for them to be maintained like equally compacted. Then acetylation has to be reduced. So when I'm creating this mutation, definitely other modifications are in place. So you should not expect and you can't expect. In yeast system, still people have shown if you remove the phosphorylation, there it's not this. I mean, there it's more, let's say, black and white. So there you see defects there and then. But here there are other systems. Other major player here is condensins. So condensins, they form these chromosomes, and if they are fine, then you will still see the chromosomes forming, and everything will be just okay. Maybe not at local
Sounds
scale,
good.
but yeah.
Sounds good. Sweetie, the
Yeah.
connection is not that great, so maybe we'll
Yeah.
take a quick suggestion, feedback question from Aarti, and then
Right.
we may move on. Thank you, Kiran. Thank you, Sweetie. Just the connection is not that great.
Yeah.
Yeah. Yeah.
So, in the interest of keeping it quick, I would say, sweetie, great work, but I feel I just felt like I was not able to understand exactly what experiments were done. So often the question was framed as the title of the presentation, and I understand the high C data and all, mostly because I'm familiar with Dimple's work. But I just I could not figure out what was the data being shown after apart from the high C experiment, which was obvious. So you might want to work on explaining the experiment first. The other thing is, very broadly, you started the whole thing with saying you want to figure out what helps to keep a bookmark of the interface signature, right? The whole seminar seemed to be framed around this question that there is some sort of memory, and that bookmarking brings a cell back to that state, or at least that bookmarking of tags and genes has to be remembered.
Right.
I didn't feel like you answered that question till the end. I understand that for some reason you picked H3S10P, which you might be better off explaining how you narrowed into that one protein anyway. But let's say you narrowed into it for whatever reason. But at the end of it, what you have shown is the correlation between that protein, its association with the active domains, etc., 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 which was great. I still don't understand why would that be a bookmark. I mean, isn't that everything that's temporally and spatially regulated, whether it's clock proteins or any other transcription factor, that periodicity can't be called bookmarking, right?
Right. So bookmarking is essentially when you have something in interface, then in mitosis you retain few certain levels of it. You lose most of it, but you retain certain levels. So that is essentially bookmarking. In our study, we are not calling H3S10P as a mitotic bookmarker. What we are trying to say is that H3S10P, which is increasing in mitosis, is plainly sitting where the domains were, and therefore the cell has a mechanism in place where physically the domains are absent, because if you do high C you don't see them, but something is sitting there to hold that place for the domains to come back. So like you said, we are not like fully able to say, so if you go back and see the title of the paper, we are not able to say it fully that this is actually the domain marker, but we are still able to conclude that it has some role in keeping those domains compact, and therefore if you don't keep them now you will have to do in the mutants maybe another round of high Cs and other experiments, but yeah, that's there. So we are sort of saying that it's physically marking it and having
Okay, so just because not everybody will go read the paper, I would suggest in light of your data, you should reframe the question you pose in the beginning. Otherwise, it seems to fall short. Okay, otherwise, great job.
Yeah, thank you.
Great feedback. Thank you. Thank you. This is very engaging. I hope Sweetie and Satyam have both felt that the mental feedback and suggestions have been very engaging. Thank you, all three of you, and thank you for your talk, Sweetie. Well done.
Thank you so much.
Take care. Bye-bye. Bye. All right. Our next speaker is Debraj. Welcome, Debraj. And
Thank
we
you.
see you. We hear you. You can go ahead and share your slides.
Sure.
Debraj, you have 20 minutes, starting now.
Yeah, thank you. Okay. So, hello everyone. So I'm Debraj and I am from Professor Sandeep Mishra's lab at IIC Bangalore. And today I'll be talking about regulation of the expression of ribosomal proteins by small downstream ORFs. So to start with this, so the genetic information in our cells passed from the DNA and it is copied into RNAs, and some of it is copied into mRNAs. And these mRNAs are then read by the ribosomes, which are these ribonucleoprotein complexes that give rise to the proteins, which has many functions in our bodies. Now, these mRNAs have mRNAs in eukaryotic cells mostly have this structure where they have a 5' cap Followed by it is an untranslated region, often called, always called the 5' UTR. And then there is the CDS or the coding sequence, which has a start and stop codon for initiation and termination of translation. Followed by it is the 3' untranslated region, another untranslated region, which is, and after this is the poly 8A. Now, because we focus on translation in the 3' UTR, so with time, although these regions were called as untranslated, with more high throughput studies, it has been seen that ribosomes do occupy these regions, and often these regions also get translated. Now, because we focus on translation in the 3' UTR, I will talk about only this. And translation in the 3' UTR can be of two different types, either stop codon read through, where ribosomes that are translating the coding sequence reads through the stop codon and continues translation beyond the stop codon and only stops at next in-frame stop codon that it encounters in the 3' UTR, thereby giving rise to a C-terminally extended protein isoform. And this phenomenon is stop codon read through. However, in case of translation reinitiation, there could be initiation and termination of translation right at the 3' UTR. And so this protein will not have any parts of the CDS. It will be translated directly from the 3' UTR. So how do you really find out about translation going on in the 3' UTR or for for the entire mRNA? So one way of doing this is to analyze ribosome profiling data. And ribosome profiling data essentially gives us the fragments of mRNAs where ribosomes are bound. So they are mapped back to the transcriptome, and that's why we know that these positions are getting translated. Now, if there is ribosomes binding to the 3' UTR, that could be a good indication that translation could be going on over there. Now, for our data, what we did was to retrieve the publicly available riboseq data from SRA database of NCBI. Followed by it, we aligned the reads from all transcripts for all studies for every transcript. And then we calculated the number of reads per nucleotide. And this gave us the reads density for the CDS, the proximal 3' UTR, and the rest of the 3' UTR. And then we calculated the average reads density per transcript from all studies. So this could be a little confusing. So here is the data for this. So here we have the average reads density of the CDS, the proximal 3' UTR, and the rest of the 3' UTR. Now, looking at these ribosomal protein transcripts, you can see for RPS27A and RPL36A, although the average reads density in the CDS is more or less similar, and it is high, which is expected, in case as compared to the other ribosomal protein transcripts, you can see in the proximal 3' UTR, the average reads density is significantly higher as compared to the other ribosomal protein transcripts. And later, if you see the rest of the 3' UTR, it's actually more or less similar to all ribosomal protein transcripts, suggesting that RPS27A and RPL36A could undergo translation in the proximal 3' UTR. Now, although I have worked on both S27A and L36A, for this presentation, I will focus on L36A primarily. L36A or RPL36A is also known as EL42 and is a component of the large subunit of the ribosome. It shares sequence similarity with RPL44 in yeast and belongs to RPL44E or RPL36AE family of ribosomal proteins. Now, so we were looking at ribosome footprints, right? So here is the data. And if you look here, this is from RiboCrypt, and this shows ribosome footprints in the CDS or the coding sequence. But if we zoom in over a portion over here, we can see that this being the canonical stop codon, followed by it is the entire 3' UTR. And this is only a part of the 3' UTR. And here you can see that this is a start codon in the 3' UTR, which also has an in-frame stop codon in the 3' UTR, thereby giving rise to a putative small ORF. And this small ORF is downstream to the main canonical ORF, so we call it a small downstream ORF. And if you see the ribosome footprint over there, here in this frame, this also increases with the start codon and falls down after the stop codon. And this codes for 6 amino acids and is a 7 codon ORF. Now we wanted to validate our results with experiments. So we cloned this entire coding sequence of L36A with its start and stop codon. Followed by it is a region which is present in between the main ORF and the downstream ORF, and we call it the internal UTR. So followed by it is the downstream ORF. The downstream ORF either has its start codon or has the start codon mutated. Followed by it, for now, it's F-loop only, which is firefly luciferase. We'll come to GFP later. So transfecting these constructs in HEK293 cells, we saw that when the entire sequence was present of the downstream ORF, we could see that translation was actually going on in the downstream ORF, which is seen by the relative luciferase activity of F-loop. And this was not seen when the downstream ORF was not present. And this relative luciferase activity was also significantly higher as compared to when the start codon of the downstream ORF was mutated. Similarly, with the GFP-HA constructs, we saw that when the GFP was present, and in both cases, FLUC and GFP doesn't have their start codon, so translation can only initiate from here. And we saw that GFP expression was seen when the downstream ORF was present, rather than when the ATG was mutated. Similarly, in case of Western blot, we saw that when the ATG of the downstream ORF was present, we could see the reinitiation band, which was not observed when it was the ATG was mutated, suggesting that downstream ORF of L36A is translated by independent initiation. Now, to check for the importance of this downstream ORF, we looked through all different mammals and whether this sequence is conserved across mammals. This is the canonical stop codon, this is the start codon of the downstream ORF, and this is the stop codon of the downstream ORF. And we saw that the downstream ORF's position was thoroughly conserved across mammals, and in most cases, even the sequence was conserved. So we asked, what would be the mechanism of translation initiation in this downstream ORF? And now the easiest thing that would come to mind is IRES-mediated translation initiation, which is translation initiation at an internal ribosome entry site on the mRNA. We checked that, however, that was not happening in our case. So we wanted to check whether translation at the downstream ORF was actually dependent on the translation of the main canonical ORF. And for this, we cloned a staple hairpin in between the main canonical ORF and the internal UTR downstream ORF region. So again, and followed by this is firefly luciferase without AUG. Now, if there is no recruitment of the ribosome directly over here, translation at the downstream ORF should decrease over here if these ribosomes are actually going on to translate here because of this steric hindrance. And that's exactly what we saw in this wild type sequence as compared to this. When we put the hairpin in between, we saw that translation is coming down. And we wanted to make sure that this is actually happening. For this, we cloned this hairpin just at the 5' end of the main canonical ORF so that translation does not even initiate from here. And then this downstream ORS translation would also be impacted from our previous study. So that's exactly what we saw. In the wild type sequence as compared to this one, in the hair, when we inserted the hairpin, translation was significantly lower as compared to the seen by the relative luciferase activity. And we also checked with a control sequence which we placed instead of this hairpin of the same length, which does not form any structure. And we saw that the relative luciferase activity was comparable to that of the wild type sequence, suggesting that translation initiation at the downstream ORF is dependent on the main canonical ORF. Now, what could be the importance of this translated downstream ORF? This is a very small ORF. So around the time when we were doing this study, another paper came up which said that translation of small downstream ORFs enhances translation of canonical main open reading frames. And this got us intrigued. So we wanted to check whether this is actually happening in our case. And for that, we designed this full-length construct of L36A, where the entire coding sequence, the internal UTR downstream ORF, and the entire 3' UTR is present. Flag HA is kept at the end terminus of L36A such that we can detect L36A protein. And either the start codon of the downstream ORF is present or it is mutated. When the start codon of the downstream ORF was present, we saw that the protein expression, the L36A protein expression was significantly higher as compared to when the start codon was mutated. This has been quantified over here. And we wanted to check what is actually going on with the RNA levels. And we saw that even in the RNA levels, when the start codon of the downstream RNA was present, the RNA levels were significantly higher as compared to when the start codon of the downstream RNA was mutated. So how does the translation in the downstream RF affect the mRNA levels? And there could be multiple ways mRNA levels can be regulated. And one such way is alternative polyadenylation, where you get different lengths of mRNA and they have different stability. And we checked that. And that wasn't happening in our case. So the next thing that we caught up on was that when we checked this sequence of the downstream RF, we saw multiple microRNA binding sites overlapping with the downstream RF. And we wanted to check whether this could be regulating this mRNA levels. And for that, because these microRNA sites are overlapping, we mutated these three nucleotides such that all of these three microRNAs can no longer bind and no new microRNAs are introduced as well. And with this, we mutated this in the mutant downstream RF background where we were actually seeing less RNA. And then we checked the RNA levels and we saw that when the downstream RF was present in the wild type sequence, we had high RNA levels. And when the downstream RF was mutated, as we have seen, the RNA levels were low. But when this mutation of microRNA binding sites were done in the mutant background, we saw that RNA levels were rescued again as compared to the wild type sequence. So suggesting that microRNA-mediated regulation of L36A could be reversed by translation in the downstream RF. However, this is just a preliminary result and we are following up on this. Now, does this also happen in the endogenous context? To check that, we deleted the downstream ORF region in HeLa cells using CRISPR-Cas9-based technique. And we checked the L36A mRNA level in these cells. And we found that in the downstream RF deletion clones, the L36A mRNA levels were also significantly lower as compared to the wild type HeLa cells. And similarly, the protein levels of L36A were also significantly lower as compared to the wild type cells. And this is exactly what we see in our overexpression backgrounds as well. Now, considering that this is a ribosomal protein and ribosomal protein's expression is going down, this could also mean that there could be an effect in global protein translation. And we wanted to check that. So to do that, we did a ribopyromethylation assay where we can detect global protein translation levels. And we saw that in the downstream mode of deletion clones, global protein translation was significantly lower as compared to the wild type HeLa cells. And we also checked proliferation status of the cells, and we saw that the deletion clones had slower proliferation rate as compared to that of the wild type HeLa cells. Now, I was talking about L36A for this time, and we also saw similar results in case of S27A, and this is just a very small compiled result where we see that with the flu construct, we saw that when the downstream ORF was not present, we saw significantly lower relative luciferase activity as compared to the wild type sequence. Similarly, with GFP-HA, we saw GFP expression only when the ATG of the downstream ORF was present. And we also saw the band of reinitiation only when the ATG of the downstream ORF was present as compared to when it was mutated. Similarly, with full-length constructs, we saw that only when the downstream ORF's ATG was present, we could see higher S27A protein expression and also higher S27A mRNA expression. So in all, for the L36A study, when the downstream ORF is present, this region is so ribosomes can actually bind to this region, occupy this region. So microRNA risk complex cannot bind to this place. So normal L36A protein levels are formed. This leads to normal number of ribosomes and normal global protein translation and cell proliferation. However, when the downstream ORF was not present or the start codon of the downstream ORF was mutated, ribosomes do not occupy this region and microRNA risk complex can now bind to this region. This leads to translation repression and mRNA degradation. This also leads to reduced L36A protein and RNA levels and reduced number of ribosomes, which causes reduced global protein translation and cell proliferation. So finally, I would like to thank all my lab members and my supervisor, Professor Sandeep Mishwapa, and IASC and all the funding agencies. And definitely Karishma and the entire team of India Bioscience and the company of biologists for providing this opportunity for PhD students to present their work and providing me the opportunity to present my work. Thank you.
Thank you, Debraj. Great talk. So Aarti, Ram, and Kiran can take it forward for 10 minutes of Q&A.
Start? Yeah. Nice work, Devraj. Just want to know, like, you really need that additional start codon because if it is that ribosome is there to begin with in the main transcript, right? So somehow it seems that that stop codon, which is canonical, is being skipped, or it is independent read because identifying that additional start codon seems to be important, right?
Yeah, so we have also seen that when the start codon is not present, so it's not a read through. So the ribosomes are not bypassing the canonical stop codon. It's just that the ribosomes are initiating fresh in the 3' UTR on the start codon of the small downstream OR. So, I mean, I have not shown multiple experiments, but we see this thoroughly that this is what is happening and there is no read through. The canonical stop codon is not skipped.
So you really that internal UTR which you talk about, that's important for the fresh ribosome binding towards that new start codon. Is that important for ribosome holding?
Yeah, so what we see is that the internal ribosome, internal UTR
UTR.
is actually important in case of translation at the downstream ORF. When it's not present, translation comes down. However, it does not provide an internal ribosome entry site to the ribosomes. So what we think is that the 60ase is probably just after termination at the canonical stop codon, the 60ase is moving towards the downstream ORF. And then again, the reassembly of the large subunit and the small subunit happens and translation initiates from the small downstream ORF. Yeah.
Okay. So basically, this is something very specific to ribosomal proteins, or it is a general phenomena? And also, is there a correlation between the type of canonical stop codon? Because I think the image which you showed a comparison, all were TA as a stop codon, if I'm not wrong. So is there any
Right.
correlation with that?
Yeah, I don't think there are any correlations with that because there have been high throughput studies where they have found evidence that I mean, downstream ORFs are getting translated, but there aren't any candidates that have been characterized fully for the downstream ORFs peptide or anything. And these downstream ORFs are present across multiple transcripts, not just ribosomal protein transcripts. But here in our study, we are studying ribosomal protein transcripts.
And any idea, any property of these small peptides which you are generating from this DORFs?
Right. So we are testing with the peptides right now whether these peptides actually have a functional role in the cells. And this would be like very soon to, I mean, very preliminary studies to talk about. So I will hold on to it and we'll talk about it later.
Okay.
Yeah, Devraj, nice talk. I just want to ask you, are there any full length L36A that you can see in the that is part of the ribosome? Full length meaning the downstream ORF, whatever it is, some 6-7 amino acids, right? That being
Right.
part of full length. If there is read through, would the function
No.
of that L36A would be different or yeah.
Right. So read through is not at all happening. We have tested by multiple experiments, and so the initiation is independent to that of L6 CDS. So downstream ORF has an independent initiation, and these are not fused proteins. So the downstream ORF's peptide is translated separately, and it is not.
Okay. But what would happen if you fuse this peptide to L36A? Would your L36A would still be functional?
I am not really sure because even in the wild type sequence, it's not fused to L36A. So I don't know what will really happen. Yeah.
Okay. And as Ram was asking, so is this is not confined to just this particular ribosomal protein, right? It is kind of universally you see the presence of downstream ORFs.
Yes, across many transcripts, downstream ORFs are present, and we are just studying these transcripts. But just like upstream ORFs, these are present across multiple transcripts.
So, is there any kind of bias you see in the GC content or AT content of this particular segment, this part of the gene? Because these downstream ORFs are a feature of some parasites, right, which are like AT genomes. There are multiple start sites. For example, if you look at falciparum and Toxoplasma gondii, for example, there are these multiple start sites, and they have some regulatory role in the expression of the ORF itself. And there are also those short peptides are made as well.
Right.
Yeah.
So, given that the downstream ORFs are present in a 3' UTR and 3' UTRs being rich in AT, that's the only thing that we see for now. But then we also did not really go on and check all downstream ORFs and see if there is a bias. So, yeah, we can do that.
So the last question,
Sure.
L36A is constitutively expressed, right? I mean, it has to be.
Yes,
Is
it is a
do
core
you
ribosomal
see-
protein. It is constitutively expressed.
So in the absence of L36A, you are saying ribosome is not functional, or does it have some defect, or how does it? Because even there are a lot of
Yeah,
redundancy in the function, right? Yeah.
right. But there are defects in global protein translation and even proliferation. So there have been studies where people have shown that, I mean, how if L36A is mutated or it's not expressed, what happens?
Okay. Okay, Devraj, great. Thanks. Karishma, maybe
Thank
you
you.
can. Thanks.
Hi.
Hello.
Can you hear me?
Yeah, yeah.
Sorry. So it will sound like my question is redundant with what has been asked, but I'll still ask it in a different way. First of all, excellent work. And I found
Thank you.
this to be a very lucid presentation. Thanks. So I'm still wondering, like, has the natural the protein, has this been detected in cells apart from the experiments you have talked about? I'm just curious to know, with a 6 amino acid
Right.
peptide,
So,
has this been detected? Because it's one thing to add a whole giant reporter at the back end of this protein and to show that it can be made, and it's a
mm,
whole different thing to know that the cell is actually making it. So what are your thoughts on
right.
that?
Right. The thing is that because it is a very small peptide, it's a 6 amino acid peptide. It hasn't been detected yet with mass spec data or anything like that. If you're asking about that, but then there are multiple evidence from this ribosome profiling data, and there was a recent paper in 2024 in Nature Communications which also showed that the evidence of this peptide being formed in the cells, but obviously it was not a mass spec analysis. Yeah.
Right. And okay. And so that the other question I had was, so now this is the mRNA which is encoding a ribosomal protein, uh,
Mm.
right?
Yes.
So, it, is has it is it isn't there some understanding that ribosomal protein, I mean, would the mRNAs of ribosomal proteins, do they show a higher propensity for ribosome binding in the ribosome profiling experiments, simply
Yeah.
by virtue of the fact that you need to make a boatload of ribosomal proteins? Aren't they known to
Mm.
show a lot of ribosomal footprints on them?
You would say that they being translated more, you would see
Yeah,
more footprints.
yeah.
Uh, I I am not really sure, but I don't. I think for the rest of the 3' UTR also, we do not really see ribosome footprints in the rest of the 3' UTR. However, we see an enrichment only at the downstream ORF's position. So I would think that that says that specifically at the downstream ORF translation is going on.
Okay. And my last question is, it's sort of along the same lines of being able to see this, see whether this peptide is made actually in cells. Would it help to combine your ribosome profiling work with sort of polysome information, like finding ribosome profiling is not telling you that something is actively translated. It is telling you that the ribosome is bound, is found bound to that that RNA, right? So is there some such information out
actually,
there?
thank you. That's a great suggestion. And we are actually doing that. So we'll be doing that polysome assay to actually check whether ribosomes are bound there. And we can show that also as an evidence saying that here ribosomes are binding. Yeah.
And whether they move in and out of polysome fractions.
Yeah, we'll be doing that.
Okay, thank you. Very good.
Thank you.
Yeah, if I can ask Karishma one small question.
Sure, Ram. We have a minute or
You
two. Yes.
suggested that this presence of DORF actually helps in the mRNA expression, right?
Yes, in our case, yes.
Yeah, and also it's like
Translation in the downstream order.
Translation. Also, so and it is part of your ribosomal mRNAs, which are proteins which are in the ribosomes, right? RPL36 and 27A. So what I'm trying to get to is that is it like that some of these housekeeping genes which will require more expression can have this signature attached to their 3' UTR region so that they're expressed more and their more protein is made?
Could be. I would rather think that it could be a regulatory mechanism. So probably under certain kinds of cues, this downstream ORFs getting translated will actually make some transcripts or some proteins from some transcripts get translated more. And that's something no one has shown us yet. And we are trying to find out with our microRNA based experiments whether this is happening with our case, and probably that could give an unique mode of regulation for ribosomal protein transcripts is what we are hoping.
Good. Okay.
Thank you.
Well, thank you, Debraj. Wonderful talk. Thanks. And to our expert mentors, the feedback is so detailed. Thank you so much. This really, like, we are talking at India Bioscience, this seems like a defense. We hope you have a pre-defense experience. Great. Thanks, Debraj. Our next speaker is right here, Ajit Kumar. Welcome, Ajit. We can see you. Put your video on, or your audio on, and yeah, we can see your slides too.
Yeah.
So, Ajit, your 20 minutes starts now. Okay?
Okay, so,
all the best.
Thank you, everyone. I'm Ajit from CSR IGIB, PhD student, and working in the field of structural biology particularly. And my title of talk is Structural Insight of CRE, which is a cis-secting replication element of stem loop 2 and its interaction with UP1 domain of HNRPM protein in regulating its hepatitis C virus replication. So coming to the its brief introduction. So hepatitis C virus genome have around 9.1 kb long RNA, positive standard RNA, and it cause like hepatocellular carcinoma and the liver inflammation. And the 5' and 3' end of its genome consist of various conserved structural elements, and these elements is shown to regulate its translation and replication. So one paper, one study came from a Nobel laureate lab, Charles M. Rice. He explained that the long-range RNA-RNA interaction between these conserved conserved structure of these 5' and 3' end is shown to regulate its replication, basically. So one long-range RNA interaction is well studied by his lab is long-range interaction between this CRE region, which is its replication elements. So this element consists of 3 stem loop structure known as 5 basal 3.1, 1, 2, and 3. So they've shown that the long-range interaction between this stem loop, that is 5 basal 3.2, and this stem loop 2 at the 3' end it makes the kissing loop kind of interaction, and any perturbation in this kissing loop interaction lead to the reduced replication of this hepatitis C virus. So overall, his study shown that this long-range RNA-RNA interaction of HCV genome are essential for its replication. So, there is an extensive study which is talking about that how this long-range RNA is actually regulating the its HCV replication, but there is not many studies which talking about the host factors, host binding proteins, which are regulating its replication and translation process. So one study came in 2016 in scientific report, which they wanted to in some host interacting protein, which is particularly binding to that C-secting replication elements. So he did pull down study, RNA pull down study, and followed by the mass spectroscopy. They found like a few of proteins which is particularly binding to this CRE region. And out of those protein, he also validated through in cellular work. They did like knockdown and overexpression study. So out of these proteins, they found that HNRP-A1 and HMGB1 is found to be a negative regulator of its HCV replication. And out of these two proteins, HNRP-A1 is found to be a more critical of to regulate viral replication, as it's shown by various other studies where HNRP-A1 is also regulating the viral replication in case of other viruses as well. So this CRE, as I explained earlier, it has 3 stem lobe structure, 5 basal 3.1, and 3 is shown to regulate translation process. This study again came from same lab. They explained they studied the essentiality of these 2 stem loop structure in the translation process, but 5 basal 3.2 stem loop structure is shown to regulate the replication process of its HCV virus. So here I have taken model for to study the molecular mechanism of interaction between HNRP-A1 protein and this stem loop structure, which is 5-PSL 3.2. So coming to interaction study, first I wanted to see the evolutionary conservation of this 5-basal 3.2 stem loop structure. So I have retrieved around thousands of sequence from the RNA viruses database, and from there are various subtypes of this HCV virus. So from each subtype, I have included around 100 sequences, and the sequence of 5' basal 3.2 were aligned and made the consensus sequence. And we, from this phylogenetic analysis, we found that the region which is found to be sequentially conserved is also critical for the long-range RNA-RNA interaction. So as the sequence is derives its structure, so I also folded the consensus sequence from each subtypes and found that this 5' basal 3.2 structure is also conserved, and it is folding in stem loop structure, consists of the 12 nucleotide apical loop and 8 nucleotide bulbs, and bulbs is separated by 2 base paired region. So we wanted to see how the low resolution structure from UV melting experiment. So UV melting experiment basically give the information about the base pairing regions. So I did the UV melting study from 20 to 100 degrees Celsius. And we indeed found 2 inflection points suggesting that this this RNA is actually folding in somehow similar way, giving the two transition which is coming from 2 base paired region of this RNA. So overall, this study suggests that 5 basal 3.2 forms a stem loop structure that is conserved and thermodynamically stable. So here we concluded that yes, it is conserved and thermodynamically stable. So next we wanted to did the interaction study with HNP1 protein. So HNP1 protein is multi-domain protein having the two RNA recognition motif followed by the RGG box and the nuclear localization signal. So to see the contribution of each domain of this protein, we did various truncation where we included the both RRM followed by the RGG domain and only tandem RRMs, which is also known as UP1 domain of this HNP1 protein and the individual RNA recognition motifs. So all constructs were cloned in PET28A vector and overexpressed in E. coli, and protein were purified through affinity chromatography and used and followed by the size exclusion chromatography and used in further study. So first of all, I did electrophoretic mobility safety assay to see whether this HNP1 protein is binding to this 5' basal 3.2 RNA. So here the RNA concentration was kept constant and the protein from 0 to 2 micromolar concentration were titrated, and from the bound fraction was plotted against its concentration, and which this slope was fitted into hill slope, which give the dissociation constant of around 644 nanomolar. So next experiment where I deleted this RGG domain and included only RNA recognition motif, which is UP1 domain of HNP1 protein. And again, the KD was extracted from this bound fraction and which give the KD around 523 nanomolar. So comparing the KD of these two constructs, we found that within the uncertainty, we are getting around similar affinity for the both constructs, suggesting that RGG box is not showing the minimal contribution in the recognition of this 5' basal 3.2 RNA, and only this region, which is tandem RRMs, RNM1, RRM2, of which is also known as UP1 domain, is sufficient for the binding to its 5' basal 3.2 RNA. So as IMSA is a semi-quantitative technique, so to get the accurate parameter of this binding interaction, we did isothermal titration calorimetry, which give the accurate binding affinity along with its deriving force of its interaction. So here again, both construct UP1 along with RGG box, and only UP1 was titrated over this 5' basal 3.2 RNA. And again, we found 1:1 stoichiometry, suggesting that one protein is binding to this one RNA. And we also again getting the similar, around similar binding affinity, suggesting indeed RGG box of HNP1 protein is showing minimal contribution, and UP1 domain of HNP1 protein is sufficient for its interaction. And as so, ITC also give the deriving force to also give the driving forces of in its interaction. So here, this interaction is particularly enthalpically driven, as seen by the negative delta H, and is spontaneous at room temperature. It is as seen by this negative delta G. So here we found, yes, UP1 is sufficient for binding of this 5' basal 3.2 RNA. So this UP1 again consists of two RNA recognition motifs. So we again wanted to see like how this individual domain of this protein is binding to its RNA. As various studies have shown that these individual RRMs have differential binding affinity for different like sequence and structure of RNA. So again, we titrated the individual RRMs, RRM1 and RRM2, and we got the reduced binding affinity, around 10-fold reduction in binding affinity, going from around 250 nanomolar to 2 micromolar and 2.5 micromolar, suggesting that both RRMs are required to bind to this like stem loop structure, which is 5' basal 3.2 RNA. And both RRM is giving its high affinity, making this high affinity interaction complex. So various truncation and sequence-based mutation were made of this RNA to decipher the actual interacting site within this full-length stem loop structure. And we found this apical loop is sufficient for UP1 binding. So here I am showing you where the UP1 titrated over the this apical loop only, and which gives the affinity around again 250 and 200, around 200 nanomolar and 1:1 stoichiometry, suggesting that UP1 protein is binding to its this apical loop, apical stem loop structure. So as this HNRP is like shown to bind with the single standard region of RNA. So I took this single standard region of this apical loop of this apical stem loop structure, and again UP1 protein were titrated over the linearized this 12 nucleotide apical loop, and we found again around 200 nanomolar affinity, and which suggests that this apical loop of 5' basal 3.2 act as a sequence-weight binding site for the HNRP1 protein. Again, this interaction was again enthalpically driven. So overall, this ITC and IMSA data suggest that yes, this is the one of the binding site of HNRP1 protein. So as this sequence and structure is so as the sequence of this RNA is derives this interaction, so we wanted to get the high resolution structure of this stem loop structure. So for that, I used the NMR as a tool to validate the structure of this stem loop structure, which was predicted through in silico tool, which is RNA-fold software. So here I am showing you a data of 1H Noji spectrum of immunoprotons, which give the information about the like hydrogen bonding information of the stem region of the RNA. So in the NMR for the base pairing pattern, it's there a defined region for the each base pairing nucleotide base pairing. So from 12 to 13 ppm gives the information about the particularly GC base pair, and the upfield of 12 is give the information about the non-canonical base pair, and from 13.5 to 15 give the information about the U with A base pairing. So here you can see like this spin system is belongs to this G with C base pairing, which is close proximity with U with A base pairing. So it give the it should see the it should see the U with A base pair. So here you can see the connection of this NOEs to U24 spin system. So again, this U24 is again in close proximity with GC base pair, which is G4. So this G4 is again giving the NOE to the G4 spin system, and again G4 spin system is in proximity with the U22. So you can see this spin system is again giving the cross NOEs to this U22 spin system. So this is how the entire work pattern was defined, and overall this data suggests that this stem region of this RNA is actually base pair, and it's folding in the similar pattern which was predicted through the RNA-fold software. So also the to assign the sequential walk of the each nucleotide, the D2O-NOG like D2O-NOG spectrum for the non-exchangeable proton also were collected, and each of the nucleotide were assigned. So here I am not showing that data due to this time constraints. So here I am showing you the data that so in ITC data we were found that this apical loop of this RNA was actually acting as a binding interface for the UP1 protein, and particularly adenines were the main interacting site for UP1 protein. So adenines of the this system loop structure were labeled through carbon 30, and the C8 and H8 of adenines were collected, and NOE for these C8 and H8 were collected, and the protein were titrated over. So from the chemical state perturbation, we can conclude that adenines were indeed adenine were binding acting as a binding site for the UP1 domain of HNRP1 protein. So again, in contrast, we also label the cytosine of this stem loop structure, and again the UP1 protein were titrated. We didn't see any chemical shift perturbation of these in cytosine, suggesting that indeed adenines of the apical stem loop of 5' base L3 point are involved with the UP1 interaction. So here we found that yes, we got to know that adenines are the main binding sites for the UP1 protein, but what residue of this UP1 protein is actually interacting with this 5' base stem loop, apical stem loop of 5' base 3.2 RNA. So for that, again, protein N15 protein were made and HSQC data were collected. And here I titrated the 12 nucleotide linear apical loop, and we measured the chemical shape perturbation for each residue, and the chemical shape perturbation deviation bar plot were plotted, and you can see again it's validating the ITC data, which suggesting that residue from the both RRMs, RRM1 and RRM2, are involved with the 5' base 5' base 3.2 RNA binding. So in previous slide, we shown that CM Rice had shown a study that this 5' base 3.2 was making the casing loop kind of interaction with 3' stem loop structure at the 3' UTR region, and this casing loop interaction was crucial for its replication. So as we found from our biophysical and biochemical data, this apical loop is actually acting as a binding interface for the UP1 protein. So we just wanted to see that what is the effect of UP1 protein on these interaction of these two stem loop structures. So for that, we did simple microscopic thermophoresis assay, where binding affinity of these two RNA were measured over the increasing the concentration of protein. So here first I did measure the binding affinity of this 5' base 3.2 RNA and X-tail stem loop structure, which was giving 589 nanomolar, and UP1 binding around 746 nanomolar. But when we made the 1:2 complex of UP1 and this 5' base 3.2 RNA, we saw the reduced binding affinity, which was going from 589 nanomolar to 3.93, around 4 micromolar. And we again increased the protein concentration with this 5' base 3.2 RNA. We again see further decrease in the binding affinity of these two stem loop structure, which was again going around 8 micromolar. So overall,
Ajit, really sorry to interrupt. Really sorry. Your talk is going really well, but your 20 minutes is up. So can you start quickly wrapping up?
yes, ma'am.
Yeah, thank you.
So here we found that HNRP1 indeed able to disrupt the kissing loop interaction in of these two stem loops in the concentration dependent manner. So from the model system, we can see that when they are making the kissing loop interaction, replication is like wild type. But when the UP1 protein coming in the picture, which disrupt the kissing loop interaction in these two stem loops, and we see the reduced binding affinity, reduced replication in HCV. So summarizing our whole talk is that UP1 domain of HNRP1 binding with this 5' base 3.2 of HCV RNA, and RGG domain is not involved in this RNA interaction, and RNA working cooperatively to recognize this RNA, and apical loop of this RNA is acting as a binding interface, and HNRP1 protein is able to disrupt the kissing loop, this long range RNA interaction in concentration dependent manner, and a deeper understanding of this system can provide a useful foundation as a small molecule intervention.
Thank you, Ajit. Very nice talk.
So I would not like to acknowledge my supervisors and Dr. Niyati and Dr. Sobik for their guidance, and my collaborators, Dr. Mandar, Benton, and Srinivas, for their help in my study. And also would like to thank Dr. Karishma ma'am and organizers to giving me this wonderful opportunity.
Thank you, Ajit. Thank you. You all were enthusiastic about it. So you brought PhD Cafe to life, all of you. So we'll move to the expert
Thank you,
mentor
Karishma.
feedback. Thank you, Ajit. Ram, Aarti, and Kiran, over to you. We have 10 minutes. Sure.
So, Ajit, I just wanted to know, like, we have already seen the interaction of EP1 with those HNRNP, A1, and the other, right, RNA. So, and then you later characterize with the NMR studies,
Yes,
the
yes,
residues which are involved in the interaction.
yes, sir.
What was the need for the ITC-based characterization? And now, in ITC, we know that you put something in the plunger, something at the receptor. So, how you have done that experiment? And why it is
So,
important to do the ITC and get the KDE and thermodynamic parameters?
So, as we all know, IMSA is a semi-quantitative technique, and which is a kind of non-equilibrium system. So, to get the accurate binding affinity, we should see the extract KD from equilibrium system. So, which is the well-suited technique, which is a isothermal titration calorimetry. So, where protein were titrated over the RNA in the adiabatic system, and the heat of interaction were measured over the course of titration. Hello?
Yeah. So
Yes,
that's
yes,
fine.
Ajit. Yeah.
That's the question I have.
Okay, sounds good.
Yeah. So, what is the physical? Sorry. Hello.
Yes, sir. Yes, sir. Can you please repeat your question, sir?
Yeah.
Yes, hi. Yeah.
What is the physiological role of this HNRNP1?
So it's a nuclear protein which is shown to regulate the mRNA stability, and it's also shown to regulate the translation process of human as well. Also, there are various studies they have shown that HNRP-A1 protein, which is a RNA binding protein, is able to regulate the replication and translation process of various viruses. But in case of HCV, it wasn't well characterized. So that's I wanted to address on my study.
I really appreciate, all the thorough characterization of the interaction of this HCV, that structure with HNRP1. But, in the context of the physiological function, what does these KD values mean? Let's say a KD of 40 nanomolar, right? What would that mean?
So, like two molecules is
No,
like
I understand. Biochemically, I understand their binding, it has very high affinity, binding is very tight. But in the context of the HCV replication, what would that binding mean? I'm not very clear.
So, here we found that that long range RNA interaction is actually perturbed by the UP1 binding. Okay. So if we will able to see strength of the binding, what the range of the strength of these two molecules are binding. On the basis of that, we can design some small peptides with the similar affinity that can hamper disrupt these interaction of these RNA and protein, and that can perturb this HCV replication.
But those peptides can also go and bind to other mRNA, right? If this protein is involved in mRNA pre-processing.
So that's why we will be doing the targeted kind of targeting from peptide to this RNA.
Okay.
So that it will not act like a full length protein, but it will be able to bind with the similar affinity which is binding this RNA.
So great presentation. Just one small suggestion. Some of your labels you had, instead of micromolar, you had U molar. which I mean, I know a lot of people here, my own student, they make all, they are a bit lazy in correcting those. So keep that in mind when you're making a presentation.
Okay.
Just
Thank you,
stay
sir,
to
for your suggestion.
micromolar. Yeah. Okay, great. Good
Thank you.
presentation.
I will improve it.
Okay, thank you.
Okay, can you hear me?
Yes,
This is
yes, ma'am.
Aarti. Yeah. So my comments would also echo some of them already made, but I wholly agree that when we hear a very nice piece of work describing biochemistry and biophysical analysis, it will still be nice to close with some perspective on biology. Right, exactly, because you took painstakingly calculated whether it's 200 nanomolar or 2 micromolar, but the implication of this to biology, it's easy enough to go and look for other proteins that might disrupt long range interactions, put them in some perspective of what is known outside or around this field when you end your study. The other thing is your thermophoresis experiment can't really figure out what was the motivation for that. So, your main point is that this interaction between this protein through whichever domains now UP1 is disrupting the long range interaction, right? That is the main conclusion that you have come to, right?
So, ma'am, this is a
Long range
only
interaction, yeah.
preliminary experiment. So I'm not claiming much on basis of this thermophoresis, but just we wanted to see that effect of this UP1 protein on this kind, like in vitro kind of, like how its UP1 concentration is affecting the like disrupting this kissing loop interaction. So that in terms
Yeah,
of
that's
definitely
I
we
think that
measure.
is an important point, because you've done many experiments to show that there is a specific interaction, which part of the protein interacts, exactly which nucleotides of the RNA interacts. So all that part was good. For the thermophoresis part, because that seems to be the only experiment where you have now added back kissing loop interactions, right? And then you are seeing whether the kissing loop interactions are disrupted by this interaction or the protein or not. So I felt a little like that seemed like very preliminary. And also,
ma'am,
it's not convincing. Sorry, may I finish? Yeah. It's also not convincing the way you have taken two molecules in trans. How do you know that the kissing interaction is happening, first of all, apart from the control mutant?
So there was a study from CM Rice lab. They have explained very well that this between the 5-BCL 3.2 and that 3-XTL RNA is making a kissing loop kind of interaction, and which is essential for its replication.
No, but did they take the same trans, the two molecules in trans? So you take the two loops in trans, right? I mean, you take two molecules of RNA, add them together with the hope that you're creating that kissing loop interaction. And then on top of that, you add the protein. So is it, I mean, since you're doing all this NMR and all of this, you should be able to know if when you add these two RNAs in trans, does it or does it not even make the kissing loop interaction, first of all? That should be very tractable given the kind of experiments you're doing. And then on top of that, if you add the protein, that interaction goes. So I would suggest like you think about it a little bit. Otherwise, the thermophoresis seemed little preliminary for a conclusion that, according to me, would be the main point of what you're trying to say. Right? You're trying, otherwise, what would be the point of exactly those adenines showing an interaction? The point would be that there are long range interactions which are disrupted, right? So just go back and think about what that thermophoresis means really, and whether you should pursue, I mean, you should think about what's happening with that kissing loop interaction. Yeah, I guess that's my main question.
Okay, ma'am.
Thanks.
I will see and
Thank
improve.
you, Aarti, Ram, and Kiran.
I just
Detailed
want to add
feedback.
something. So
Please.
to what Aarti says, actually, it will make real sense if you do in the context of how biologically it will be relevant, as well as your UP1 molecule, which you showed, has additional domains. So maybe commenting on that how those will be playing important role will be better.
Right.
Yes. Sir,
Sorry, my other question is, can I ask one more last question?
Yes.
So in the first part, you showed a lot of other interactors. I'm not very sure why you picked up, only this HNRP. But anyway, probably maybe I missed it. It is okay. You really don't have to go. We may not have time. I don't know. The first slide, you showed so many different proteins. So
Yes,
why picked only this? Probably because based on literature, right?
yes, sir.
Okay, because there are so many other
Yes,
HNRPs.
sir.
Again,
These 2-3
these are
protein were validated, and out of this protein, 2 protein was found to be negative regulator of HCV replication, and this
Yeah.
HMGB1 binding to the 5 prime of this RNA to regulate its replication.
Yeah, again, you really have to, take all these data with a pinch of salt. And I know your data is really fantastic. You just have to put it in the right context.
Okay,
Good.
thank you, sir.
Yeah.
Thank you all. Once again, really detailed feedback. Thank you, Ajit. Well done.
Thank you,
Good
ma'am.
talk. Good work. Keep up the work. Okay. Our next and final speaker for this session is Priyamvada. I see her in the meeting room. Priyamvada. Yes, I see you, Priyamvada. Great. You can put your audio on. We hear you too. That's good. And you can start your slide share. And yeah, as Priyamvada is doing that, 2 important points. The feedback form for this session is in the chat window. So keep giving us your feedback, please. And the giveaway has still to happen at the end of this session. So keep the chat window active if you want a physical copy of our latest book from India Bioscience. All right. So all the best, Priyamvada. We see your slides. Best wishes.
Thank you, ma'am. Good afternoon, all of the people who are listening there. So I am Priyamvada, working at Medical and Biological Computing Lab at Vellore Institute of Technology. So basically my work today, whatever I am presenting, is on unraveling the potential of marine compounds in colorectal cancer therapy against top-2A. It is all computational approaches which I have followed to subsidize this project. So coming to my first introduction regarding colorectal cancer, it is basically the third most leading cancer all across the world, accounting for around 10% of the total cancer which is projected. So it is one of the major issue which is seen in the aspects of cancer which has to be taken into consideration for the treatment as well as for the diagnosis purpose. So this is the data which I am showing is from International Agency for the Research on Cancer Statistics, which have shown that from 2020 to 2040, the incidence rate regarding colorectal cancer will increase to 56% and the death rate will increase to 69%. Here we can see it is the top 10 countries which is being highlighting the condition, whether it is in the terms of incidence rate as well as in the terms of death rate, that in the coming 20 years, this colorectal cancer is going to be having more effect on the population. Here we can see that the incidence rate is almost same in the developed as well as the developing countries. But when we see the death rate, it is seen that in developed countries, the death rate is less. Might be the possibility why it is like, I think so, the diagnostic process, what they have is better than developing countries, as well as the treatment strategies, what they are following is better. So coming to the research gap, what I objected was like there is a lack of and the biomarker choices available for the diagnosis of colorectal cancer is limited, and whatever it is there, it is giving high false positive rates. And second thing, what was I was focusing on was the dual function of the top 2A, that is basically DNA topoisomerase. When we are talking about dual function, we are focusing on it acting it as a biomarker as well as it can be targeted. So we can call top 2A as the therapeutic biomarker. To address this research gap, I have objected one question, that is, which is the safe alternative ways to combat the target alteration in colorectal cancer. To address this is the workflow which I have followed to address the research question, where I have taken the transcriptomics data and we have analyzed it extensively through different in different analysis, like in the terms of identification of hub genes, how to correlate with the immune infiltrate, along with the redundancy analysis, and followed by its interaction with the different drugs. Once we identified the hub genes, we tried to understand what kind of mutation is affecting the top 2A. Followed, we tried to check those SNPs, what is its effect in protein stability, and further on, we have screened some marine compounds and we have performed the molecular docking and molecular simulation to come up to a potential drug molecule. Coming to the result section of my study, it started with, we have taken the microarray data set with from gene expression omnibus. It has data sets regarding different types of sample. So my accession ID was GSC 21815, which contained 132 samples, among which 66 were tumorigenic condition and 9 were normal. Rest were basically they were dissected, so I didn't consider those samples. These 66 were non-dissected, and we further proceeded with this differential gene expression to understand what kind of upregulation or downregulation is affecting this tumor samples. We performed the using gene expression omnibus 2R. This is our programming based tool, which help us to differentiate between the upregulated and downregulated genes. And we found that 1,106 genes were upregulated and 203 genes were downregulated. This altered genes were checked for the significance at the p-value less than 0.001 and log FC value of greater than 1 and less than -1. Once we curated the upregulated genes, where our objective was to check it for, we considered only upregulated just because we wanted to target it further on. So we took this upregulated genes and we have tried to check their interaction. That is, how well is this upregulated gene interacting among themselves. For that, we used a tool called string database, wherein we have put these upregulated genes and we have curated the interactome, and we visualize this interactome using cytoscape. There is a plugin called CytoHubba, which had different topological parameters in order to identify the hub genes. And at the end of the result, we found that there were 7 genes, make CDK1, CCNB1, ORCA, CCNG1, IL6, CCNA2, TOP2, and MAT2L1 was satisfying at least 7 parameters. So we considered these 7 genes for our further analysis. Further on, we performed the gene ontology terms and pathway enrichment, where gene ontology terms basically included 3 terms called biological process, cellular component, and molecular function. These 3 components basically included what my genes were associated with was mitotic cell checkpoints, mitotic DNA integrity checkpoints, and followed by histone kinase activity. It was because CDK kinase proteins were available there. Followed by, in the pathway analysis, we also saw that these proteins were associated with mostly with the cell cycle and cellular senescence. These proteins were further checked for their effect on the survival rate of the patients. So here we consider one parameter called hazardous ratio. Hazardous ratio, we considered that it should be 95% at least, which indicates the probability of the death due to the positive involvement of these hub genes, where we found that CDK1, CCNA2, and TOP2A MAT2L1 was highly expressed and it was involved in the overall survival rate of the patients. Coming to the redundancy analysis of the hub genes, this we did with DivMap tool. This is basically, it has 700 cancer cell lines there, wherein we can give our, we can know the effect of a gene on, they have knockout studies there and the CRISPR knockout as well as RNAi. There in which they have showed most of the, if it is 50% of the cells are showing their effect, we consider that as both diagnostic as well as their involvement during the prognosis of the cancer. So after this analysis, we got to know that CDK1, TOP2A, and MAT2 are not only diagnostic, but can be a potential therapeutic target. So coming to among these 3 genes, we had, when we checked their mutational rate, we found that TOP2A had more potential of getting mutated. It was around 3%. And many other papers also showed that this TOP2A alpha has role in different types of cancer. People have also mentioned their role in colon cancer as well as in breast cancer. Coming to the, once we found that maximum mutation in colon cancer is in top 2 among those 3, we try to identify these mutations and these non-synonymous mutations we try to identify were 24, and this top 2A protein consists of 1,531 amino acid, and among these 24 non-synonymous mutation, only 8 mutations which were present in the catalytic domain of the top 2A, we focused on that. And among 8 mutation, only 4 mutations were deleterious in nature, that they have some role in the disease causing effect. So this is the effect, we calculated the role of this mutation on the protein stability and the functionality. So when we compared the stability, we found that among these 4 mutations, only D1021Y was showing stabilizing effect with the delta delta G with 0.5 kilocalorie per mole. Delta delta G is basically help us to calculate between what is the stability during the normal condition, and when if there is a mutation, what is this stability. So the stabilizing mutation help us to leads to enhanced DNA repair, and in order they will leads to cancer cell survival and accumulation of accumulation of these cells leading to cancer. This mutation also helps in causes the chromosomal instability, leading to amplification and deletion of some tumor suppressor genes. Further on, this mutation also led to some of the papers. They have shown that TOP2A leads to chemo resistance as well. So when we try to check the effect of this mutation, we found that due to the transition of from aspartic acid to tyrosine at position to 1021, there was seen that this mutant protein was bigger than the wild type. It was more hydrophobic in nature, and it was more negative than the it was neutral as compared to the wild type residue. And due to this change, there was a loss of intra-chain polar and hydrophobic interaction, and this would have lead to the change in the dynamicity of the protein. So further on, we have curated the protein of the TOP2A from the protein data bank, and we try to induce this mutation into that protein just to check on the what is the change in the dynamicity. Further on the other side, we screen marine compounds with which is having different properties based on the Swiss ADME property. A Swiss ADME property is basically absorption, distribution, metabolism, excretion. Along with it, we have checked the toxicity also. So we have considered the reference compound here. What we have considered was PK1. It is a derivative of doxorubicin, which is the potent inhibitor of the TOP2A. And we found that it was violating the Lipinski rule with 2, and it was also having hepatotoxicity along with the nephrotoxicity. And we screened around 31,561 compounds from the comprehensive marine data bank. There is one database where we screened 31,000 compounds, and we came to top 9 compounds based on their high GI absorption, and they were not a drug property violation it was seen, and they were non-toxic. So once we come up to top 9 compounds, we tried to check their binding affinity with the top 2 and its mutant, and we found that we considered only the top 3 compounds which were showing better affinity and with least binding energy with the protein as well as the mutant protein, and we have considered for those 3 protein, we considered further analysis. That is, we performed the density function theory for those compounds. It basically tells about the difference between the electron donation of the compound. It is basically optimizing the ligand molecule and how efficiently this ligand can bind to our protein. On that analysis, we basically calculate the difference between that is basically the energy gap, the difference between the LUMO and HOMO. That is lower unoccupied molecular orbital and the higher occupied molecular orbital. The lower is the energy gap. We confirm that molecule as the most reactive molecule. Here we saw that compound 28461 was having least energy gap with 4.398, and it was more efficient than our reference compound also. So once we got this compound, we further subjected them for molecular docking analysis, where we checked the interaction profile of this screen compound. And there also we found that compound 28461 and 17683 showed a better binding affinity along with the least inhibition constant with the molecules. So binding energy of 28461 was minus 7.47 with the wild type and minus 7.62 with the mutant type. So compound 28461 and 176 was efficiently binding with the wild type as well as with the mutant type, and it was better than our reference compound as well. Further on, we performed the molecular dynamic simulation, and where we check their efficiency towards like how it is changing the dynamics of the protein upon binding, and we did this for 100 nanosecond, and we found that compound 28461 for RMSD value it shows 0.42, and 0.39, which was less than 1, and it showed that it is more stable when it is in the binding vicinity of the protein. And RMS fluctuation was also seen at the end, the C terminal of the protein. It was little fluctuating, but it was 0.2 nanomolar. It was feasible to go for this molecule 28461. And for hydrogen bond also, we can see that for the top 2A wild type protein, it was forming 3.2, like 3 hydrogen bond. It was constantly, at 3.2 is basically, we have calculated the average and we have put it, and we found that all through the 100 nanosecond, this hydrogen bond was seen with the wild type as well as the mutant type. RG is basically radius of gyration, how compact is the protein on binding of the ligand. So it was it was around 3.28, 3.27 in the both in the C case. It was almost compact. The protein was more compact on binding of the ligand. Later on, we checked for the interaction energy and SASA values. Interaction energy also was very well, like in the terms of what kind of electrostatic interaction or what kind of ionic bond it is forming. So those aspects to let us know that this compound mutant 28461 was efficient with the top two as well as the mutant type. So coming to the summary of the study, like from the first study, we have concluded that these three genes, CDK1, TOP2, and MAT2, can be a theragnostic biomarker. Along with that, we also found some of the transcription factors which can regulate these targets, and they can have, and these can also be a potential molecular signatures in the further studies. TOP2 is a potent essential enzyme which is involved in DNA replication. So we have consider that enzyme into picture, and then we have induced the mutation in the catalytic domain of the top 2A, and we try to understand its genomic what is the effect in the dynamicity of the protein. And later on, we have concluded with the carpentamide D, which is the compound 28461. It is basically alkaloid, which is helping in binding with the top 2A, and it can be potent inhibitor. So molecular dynamic simulation also confirmed that carpentamide D had lower fluctuation with increased compactness and higher interaction energy, supporting its potential inhibition. So these two work has been published in biochemical genetics and molecular biotechnology. And at last, I would like to acknowledge all my lab mates, especially my guide, Dr. Sudharamaya ma'am, Dr. Anand Anabrasu, who is my mentor, and I would like to thank Vellore Institute of Technology and my ICMR funding body. I would like to thank PhD Café to giving me an opportunity to speak in the forum. Thank you, ma'am. Thank you so much for all the listening.
Great. Let's move to the Q&A. Sure.
Yeah. Priyambada, good work,
Yes,
but
ma'am.
maybe I understood little bit, maybe because the area is very different. But what do you mean by marine compounds? What is the source of these marine compounds?
Sir, it can be any seaweeds. Basically, mine was from bacteria, which is there in the extensively. It is below the sea level, very deep it will be. And because of the stress condition, these marine compounds are very much potential to have anti-cancerous activity, and these are very less explored. Maybe due to the I could say people are not. It is isolation will be so difficult.
Okay. So you said that this D1021Y mutant is in the catalytic domain.
Yes, sir.
So have you done any biochemical experiments to suggest that how your compound is affecting the activity further on this mutation?
No, sir. Actually, the lab completely works on the computational tools and computational power. We don't have wet lab facility. I'm so sorry, sir.
Okay. So one of the compound which you mentioned was the 28641 is like carpentamide D, you said, right?
Yes, sir. Yes, sir.
And
Yes,
there's
sir.
another compound which was, I think, equally potent or good.
Yes, sir.
You have the identity of that also, and basically you wouldn't have done a biochemical assay with that also, right?
Yes, sir.
Okay. Okay, Karishma.
All right, Ram. Thank you. Kiran Aarti.
Yeah, Priyamada, good work. I have some questions.
Yes,
See,
sir.
when you
Yes, sir.
started out, right, you had only like 9 or 10 data sets or control data sets, right? From normal individual, something you mentioned.
Yes, sir.
When you started out, I think out of 132.
Yes, sir. Yes, sir. Yes, sir.
So 9 is too less, right? Did you find any SNPs in these normal data sets?
Sir, SNPs we usually check it for the any particular gene or protein wherein like we had to come to one protein because we can work on all this protein that is not
No,
like
that is okay. See, I'm just asking about your topo 2.
yes, sir. Yes, sir.
CDK, and anyway, all the replication related proteins are overexpressed, which is not
Yes.
very surprising, right? In a cancer cell,
Yes,
because
sir. Yes, sir. Yes, sir.
they
Yes, sir.
proliferate. So my question is, did you find any of the SNPs in the normal data set? I would have taken more number of, I don't know what is the meaning of normal, but I'm assuming that these are from healthy individuals.
Yes, sir. Yes, sir. Correct, sir.
Yeah,
The
so
sample
usually,
when
yeah. See, when people do these kind of studies, right, they take equal number of affected individuals and equal number of, in fact, in many cases, they take more normal samples, because when you are trying to look at the phenotype conferred by a combination of mutations, right, in certain genes.
yes, sir. Yes, sir. Yes, sir.
So there may be in isolation, those mutations can still be present in the normal population.
Normal population as well.
Yeah,
You
they
are correct,
can
sir.
also, yeah, they can predispose the individuals for some cancer at a later stage.
Yes,
So
sir. That is true, sir. It might be
yeah,
well that it might be present in the normal condition as well as in the cancer state.
that is why
But
I
we
think
need to
you
check, sir.
and also is all are any of the data sets coming from the Indian population, or is it just you have not you just took all the data set from wherever they are available?
Sir, it was not from Indian population because we were not able to get the Indian data sets. I felt like there is a lack of awareness in Indian population, sir, regarding colorectal cancer. I would say the data, because we are not very open to go and check for this because the conditions which we see in colorectal cancer is very minute, like it will be abdomen pain or something weight loss, which we don't know what is the reason behind it, and so we don't go for the treatment or the diagnosis or just for going for tests. So we don't get much of the data regarding India, sir.
Okay. And the other thing is yeah, sorry, someone has. So the
Yes,
two compounds that you picked up, right? You said they
yes, sir.
have high affinity, but they are in the micromolar range. If I
Yes, sir. Yes, sir. Yes, sir.
why do you think is micromolar high affinity? Because in the previous talk, nanomolar was high affinity. Now it is micromolar here.
Yes, sir.
So
Yes, sir.
yeah, why do you think these numbers actually matter?
And nanomolar is always better,
No, no,
sir.
no. I am not talking about that at all. So
Yes,
these
sir.
binding affinities,
Yes,
what
sir.
do they mean, right? For example, if you want to administer this compound to an individual, and this is the kind of inhibition constant,
Yes,
you
sir.
also have to look at, you probably have to take 100 times, at least 100 times more of this compound.
Yes, sir. Yes, sir. To
Because
just to deliver it to the system.
exactly, right? So would it even make sense to have a compound which has an inhibition constant in the micromolar range?
It is just like we calculate in the terms of basis of like how it is the least we take the least inhibition constant because it is like less we give the treatment is well working.
So this the other thing is this one of these two, right? 17683 is carpetamide D or something, right?
28461 is the
Ah,
carpetamide D.
so that maybe I'm wrong, but I think that is quite well established. People have actually experimentally tested that compound. If I'm not
Yes,
mistaken.
sir. Yes, sir. Yes, sir. It has to be tested in the terms of colon cancer cell lines and we have to check the efficacy, how it is working into the in vivo or in
I
vitro
think it is one
conditions.
of the very popular compounds. I don't know. I just recall.
Carpetamide A to C has been well studied, sir. Carpetamide
Okay.
D has not worked.
Okay. And again, these marine compounds, right? How did they come up with this database? Have they done some, I don't know, metabolomic kind of study from?
Yes, sir, might be, sir. They must have extracted or something like they have in around 31,000 compounds.
So which
Mostly
means
the databases regarding marine compounds are very limited, sir. And whatever data sources which is available is all licensed or paid versions. This is only one. I took all the data set what was there, sir. 31,000 compounds were there because I didn't wanted to work on a smaller scale of compounds and come up with something like. Upon this, I have another work on that is particularly like once top two A is overexpression is there. I gone through some research article where it is showing like it leads to chemo resistance as well. So when I
okay.
was going through the compounds, I came up with the camphorol, which
That's
has
okay. That's
been
okay. That's okay. So my other comment. So these databases compounds from bacteria, whatever, algal, fungal, plankton,
yes,
or
sir,
which
algae, yes,
vertical.
sir.
So just my last question. If you were to actually take one of these compounds forward, you think
Yes, sir.
again, coming back to the same question, the
Yes.
inhibition constant that you see do you think they are quite promising or not, in your opinion? Would you recommend, for example, you have highlighted this 28461 here, right, in
Yes,
this
sir,
table.
yes, sir, yes, sir.
Would you recommend somebody taking them forward for some testing?
I would not suggest if we are going for in vivo or any studies like that. I would suggest to have some compounds in our hand to carry forward the experiments, just to compare how well is this coming up. We cannot go with a single compound and we can say like this is what it is, because what the computational power does is just we screen up some compounds and we come up with two, like say we screen 100 to become to 5, and then 5 to 1 can be like, I would say it is more prominent if I say it is experimentally tested and it can come to the market.
Okay. Thanks, Primoda.
Yes, sir.
Karishma, if I can ask one question related to what Kiran was asking regarding the SNPs. So
Yes, sir, yes, sir.
I noticed in the very early slides that Chinese population has a lot of incidence rate and the death rate, right?
Yes, sir, yes, sir,
Is
yes,
there
sir.
any particular SNP associated with that and any kind of guideline for screening so that Indian population or those who, if they have happen to have some sort of SNP association?
Sir, we have to compare with the we have to compare the data sets of China and with India. Like what kind of SNPs are seen in India, whether frame shift mutation is more or missense mutation is more, what kind of mutation is more prominent in these two places.
Yeah, so
But
basically I'm asking if there's something known already from the China because the incidence rate and the death rate is very high.
sir, still now it is not there, like what kind of SNPs is more prominent in China. We have not gone through it. We have to, if we check the data sets, we can get to know, and we
That
can
would be a
come
very
up
good
with something.
biomarker initially for the predisposition and so on, right?
Yes, sir. Yes, sir. Yes, sir.
Sounds good. Okay, maybe we'll move on to Aarti. Yeah.
Thanks. So, just quickly, so from all these 30,000 or whatever compounds, how did you show us a short list? I sort of lost track of how you went to such a small number of compounds finally.
Ma'am, it is an extensive screening based on so many factors such as molecular weight. We have to check the bioavailability. We have to check the.
No, but no, I understand what you checked, but what was your criteria? Like, did you already know what is going to bind to your protein? Like,
No.
then I don't understand the basis of the shortlisting.
When we do, ma'am, the criteria what we keep is like molecular weight should be less than 500.
Why is that?
For computational, like smaller is the molecule, better is the delivery.
No, but there is no such number that is prescribed, no?
It is
Like,
in
how do you
a
come up with those
research
numbers?
paper. This all comes through research paper, ma'am. They have published and very many papers will like have a criteria like how to screen it, like based on molecular weight or GI absorption or Lipinski rule, which not violated. Then if it is any drug you want to work on with the blood-brain barrier, then it should cross or not. There are like when we do Swiss Army, there are hell lots of information they give, ma'am. In that we have to choose according to our work what we want to go for.
On my honest feedback on this is it's really hard for me to understand how, I mean, unless somebody has done this in the context of this particular cancer, I find it very hard to know how you would determine those numbers. You can look at all those numbers of those 30,000 molecules, but I really don't understand how you came to the shortlist. If there was a systematic process, then that
Yes,
is
ma'am.
very important to your work. And you should have
Yes.
taken the time to explain to us what that systematic process of shortlisting, going from 30,000 to 5 or
Nice.
whatever you
Correct.
showed us. It
Correct.
just,
Correct.
I completely missed that point. I don't think you touched upon that. Also, at various points, it's not clear to me what was the experiment you actually did to reach the conclusion that you came to. Like in the beginning of your slides, by the time you reach DFT, I understand that you did some experiments. But before that, what process you followed is really not clear to me. Maybe you should work on that. Uh,
Okay.
Computational,
Sure.
but unless you explain it to us, we won't understand. And
Yes,
also
ma'am.
for the DFT now, what is a control experiment for the DFT?
Control
Once you,
experiment to be performed with the reference compound also. Basically, it is for optimization of the molecule, ma'am.
right. So
Like
what is the reference compound? What are you considering the reference compound?
this is PK1, ma'am. This is a derivative of doxorubicin.
I see. And did you use that as a compound even while getting your marine list of 30,000?
Yes, ma'am. Yes, ma'am. Yes, ma'am. That was our first, like we have to take any reference compound, then only if it is better, then only we will go for it. Otherwise, we will discard it then and there. So.
Okay. And just generally, I just wanted to know what you think about. So you talked about this as a good biomarker as well as a good target for therapeutics.
Yes, ma'am. Yes, ma'am.
Is that counterintuitive? In some sense, like, is it, I mean, with something laden with mutations, usually a good target for therapeutics or?
Now when I'm saying about biomarker, ma'am, it can be either diagnostic, it can be the prognostic, or it can be therapeutic. So when I'm telling that it is a diagnostic, that means it will just help us in diagnosing, okay, fine, this person is getting this expression high, we can go and tell like, okay, fine, you have this problem.
Yeah, so
And
are you calling this a good marker for diagnostic?
yes, ma'am.
And so then how do you justify this being a good target for therapeutics?
Because it is a good because when we knock down these genes and the cancer cell lines, they have seen like they have drastic effects.
Hmm. But
So
this is the protein that's full of mutations, right?
yes, ma'am.
So how would that make for a good therapeutic? Like, would your molecules, all your molecules going to work on all the mutant versions of the protein, or
No.
how do?
We cannot like mutations. We can work on like two mutation, dual mutation. We can work on with, but we cannot say that these two mutation only can lead to this. Like two mutation
Sounds
at
good.
a
So
time.
Okay,
something to
maybe
think
these are
about.
things you can consider. Yeah, thanks.
Yeah, thank you, Aarti. Thank you all. Thank you, everyone. All right. So, Priyamada, you can stop sharing maybe. Yes, thank you. So we will now announce the winner of our giveaway, and that is Satyam Singh, who kept the chat window active. But Satyam is right now not in the meeting room, so we will get in touch with him and let him know. But thanks, Satyam, for doing your bit. Thank you so much to our mentors, Kiran, Aarti, and Ram. We appreciate the time and the input. We did take up your time, so thank you very much. And time to grab a quick lunch. For those of you who are still in the meeting room at 1 p.m. in another link, you'll have an open networking session with Snehal, who just wrapped up her PhD, and she'll be talking about how to build professional networks to fuel the next stage of your career post your PhD. So that's that. Feedback form in the chat window and See you at the open networking. And then today afternoon for our session on animal biology, where we also have 5 PhD speakers. Thank you very much, everyone. Have a good afternoon, good lunch, and see you around. Bye-bye.
Thank
Thank you.
Bye.
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 Bye.